the more we listen. As we are well aware that our efforts have no meaning without the people's trust, we transparently disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs, in an effort to create a reliable society and a safe nation. Our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bioindustry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country. We will prove our love for you by realizing the happiness of the people. Science of Love for Our People. The Ministry of Food and Drug Safety.
The MFDS is the organization under the Ministry of Food and Drug Safety to support scientific and technological endeavors for food and drug safety management policies. In recent years, our society has undergone significant changes, including the development of advanced science and technology, such as artificial intelligence and digital technology, and the rapid growth of the biohealth industry. In order to respond to these changes, the MFDS promotes risk assessment, regulatory review, test and analysis, national research and development based on advanced research facilities, equipment, and experts. With risk assessment, we evaluate the effects of hazards such as heavy metals and benzopyrene on humans when using products for human use and provide safety controls to prevent these hazards. Our review process aims to support life cycle management of safety from development to use to ensure that safe and effective medicinal products, medical devices, functional cosmetics, and dietary supplements are brought to the market. The purpose of test and analysis is to identify the quality of vaccines before distribution and ensures public safety by inspecting illegal foods and medical products. Our institute proactively responds to changes in the social environment and innovative technologies and conducts research and development according to the Food and Drug Safety Management Policy. In addition, we support training of regulatory science experts in food and medical product sectors and successful commercialization of innovative products by providing advice on strategies from R&D to manufacturing for innovative growth of the biohealth industry. In addition, MFDS has been playing a key role while working with many international organizations, including WHO, FDA, OECD, and building a global level of professional capabilities for reviewing. A healthy life starts with safety. As the national institution leading the regulatory science for foods, medicinal products, and medical devices, we will strive to become one of the best food and drug safety regulation agencies for public health and safety.
그날은 역사 속 사건이 아니다. 두려움 앞에 멈추지 않는 태도이며 포기하지 않고 나아가려는 마음이다. 그것이 지금 우리의 자세이고 모두가 기억해야 할 광복이다. 두려움에 맞서는 포기하지 않는 용기. 광복 80년. 과거가 아닌 내일을 여는 희망찬 시작. 빛나는 발걸음. 새로운 길. 광복 80년.
We study food. Because that is the people's health. We study drugs. Because that is the people's safety. We study cosmetics. Because that is the people's happiness. In order to ensure that your daily life is safe and happy, our love for you is realized through innovative science. Science of love for our people, the Ministry of Food and Drug Safety. The bigger our love, the stricter we become. As an agency that faithfully performs its duties given by the people, everything that our people use, from food and drugs to quasi-drugs and medical devices, to health-functional food, cosmetics, and sanitary products, reflects our thoroughness. In order to keep our food table safe from adulterated foods, germs and viruses, and hazardous chemical substances, we manage the safety of food ingredients according to systematic standards, conduct on-site inspections of manufacturers, and thoroughly manage the customs process for imported food to block harmful elements in the first place. Through the hazardous food sales prevention system, we also respond immediately to safety accidents after distribution. As the country's food and drug safety control tower, we make South Korea a nation that puts people's safety first. The more we care, the more we listen. As we are well aware that our efforts have no meaning without the people's trust, we transparently disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs, in an effort to create a reliable society and a safe nation. Our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bioindustry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country. We will prove our love for you by realizing the happiness of the people. Science of love for our people. The Ministry of Food and Drug Safety.
The NIFDS is the organization under the Ministry of Food and Drug Safety to support scientific and technological endeavors for food and drug safety management policies. In recent years, our society has undergone significant changes, including the development of advanced science and technology, such as artificial intelligence and digital technology, and the rapid growth of the biohealth industry. In order to respond to these changes, the NIFDS promotes risk assessment, regulatory review, test and analysis, national research and development based on advanced research facilities, equipment, and experts. With risk assessment, we evaluate the effects of hazards such as heavy metals and benzopyrene on humans when using products for human use and provide safety controls to prevent these hazards. Our review process aims to support life cycle management of safety from development to use to ensure that safe and effective medicinal products, medical devices, functional cosmetics, and dietary supplements are brought to the market. The purpose of test and analysis is to identify the quality of vaccines before distribution and ensures public safety by inspecting illegal foods and medical products. Our institute proactively responds to changes in the social environment and innovative technologies and conducts research and development according to the Food and Drug Safety Management Policy. In addition, we support training of regulatory science experts in food and medical product sectors and successful commercialization of innovative products by providing advice on strategies from R&D to manufacturing for innovative growth of the biohealth industry. In addition, NIFDS has been playing a key role while working with many international organizations, including WHO, FDA, OECD, and building a global level of professional capabilities for reviewing. A healthy life starts with safety. As the national institution leading the regulatory science for foods, medicinal products, and medical devices, we will strive to become one of the best food and drug safety regulation agencies for public health and safety.
그날은 역사 속 사건이 아니다. 두려움 앞에 멈추지 않는 태도이며 포기하지 않고 나아가려는 마음이다. 그것이 지금 우리의 자세이고 모두가 기억해야 할 광복이다. 두려움에 맞서는 포기하지 않는 용기. 광복 80년. 과거가 아닌 내일을 여는 희망찬 시작. 빛나는 발걸음. 새로운 길. 광복 80년.
We study food. Because that is the people's health. We study drugs. Because that is the people's safety. We study cosmetics. Because that is the people's happiness. in order to ensure that your daily life is safe and happy. Our love for you is realized through innovative science. Science of love for our people. The Ministry of Food and Drug Safety. The bigger our love, the stricter we become. As an agency that faithfully performs its duties given by the people, everything that our people use, from food and drugs to quasi-drugs and medical devices, to health functional food, cosmetics, and sanitary products, reflects our thoroughness. In order to keep our food table safe from adulterated foods, germs and viruses, and hazardous chemical substances, we manage the safety of food ingredients according to systematic standards, conduct on-site inspections of manufacturers, and thoroughly manage the customs process for imported food to block harmful elements in the first place. Through the hazardous food sales prevention system, we also respond immediately to safety accidents after distribution. As the country's food and drug safety control tower, we make South Korea a nation that puts people's safety first. The more we care, the more we listen. As we are well aware that our efforts have no meaning without the people's trust, we transparently disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs, in an effort to create a reliable society and a safe nation. Our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bioindustry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country. We will prove our love for you by realizing the happiness of the people. Science of love for our people. The Ministry of Food and Drug Safety.
The MFDS is the organization under the Ministry of Food and Drug Safety to support scientific and technological endeavors for food and drug safety management policies. In recent years, our society has undergone significant changes.
안녕하십니까?
Good morning. The 5th MFDS Global Conference on Foodborne Antimicrobial Resistance Day 2 will begin now. Distinguished participants at the venue and online, thank you very much for your time. Let me sum up the Day 1. Starting with the opening ceremony, we had the keynote speech of Professor Mosen Nagabi, and in session 1, we reviewed global policy and regulation trends on AMR management. And in session 2, we also reviewed policies for appropriate AMU in the food supply chain, and we had 6 presentations in total, and with a meaningful Q&A session, we had valuable time. And we also had e-poster sessions, and in total 5 posters have been presented. And this is day 2. We have 2 sessions and poster sessions as well. I hope that this is meaningful time for you. This conference is organized by the MFDS, and we live broadcast the session on the official YouTube channel of MFDS, and we will also post this video on the official website. Now we would like to begin the session 3 in earnest. The title is Genome-based resistance monitoring and introducing the latest technologies for antibiotic alternatives, and we have 4 presentations. And I would like to first introduce the chairperson for this session from the National Institute of Food and Drug Safety Evaluation of MFDS, Director In-Sun Chu, is working in the MFDS, and she is involved in the food poisoning investigation and the food safety evaluation as well. So she is participating in various evaluation and review, and currently she is the head of the national project now. Let us invite Ms. Ju with a big round of applause. Hello everyone. As introduced, my name is Ju In-sun. I'm in the Food Microbiology Division, and we are responsible for the tracing study and the surveillance, and we've been engaged in this sector for a long time. And from yesterday, I've been participating in this conference and listened to the various presentations and studied very hard. And today, I will serve as chair of session 3. It is quite burden for me, but still, I will do my best. We have 4 speakers in session 3. The title of session 3 is Genome-based resistance monitoring and introducing the latest technologies for antibiotic alternatives, and there will be 4 presentations. Let me introduce the first speaker. The first speaker is Professor Jong-chan Choi from Jeonbuk National University. He received the degree at the Jeonbuk National University, and he is currently serving as a professor. When it comes to the AMR, he conducted various studies, and he also advises various ministries of the Korean government. And for now, he is focusing on the genome-based resistance monitoring. Then, without further ado, I would like to introduce the speaker. The title is Surveillance of the Causative Agents of Foodborne Gastrointestinal Infection Diseases from One Health Perspective.
So good morning, everyone. This is the second day. It's the first session. Before doing my presentation, I hope for your understanding as I give my presentation in Korean. Okay.
So my topic is about surveillance of the causative agents of foodborne gastrointestinal infectious diseases from a One Health perspective. It's kind of broader topic, but I'll talk about Salmonella and Campylobacter 1. So this is my narrowed down topic for today's presentation. So foodborne disease. You might think it's gone. However, now we are seeing the upward trend again, or emerging trend. There are many contributing factors behind this, When you look at your left side, as you can see from this graph, from the production, it can be farms, or this can be caused in slaughterhouse and meat processing stage, and the meats are distributed or transported to retail, and finally end users will consume those meat. So this is the process how those meats are end up with our table. So from the production phase to processing centering around slaughterhouse to distribution centering around retail. So there are 3 steps. So we did sampling and we isolated those E. coli, and this is our project. So AMR requires multiple approaches. So in the previous setting, it was put much emphasis on clinical setting or in animal health. Farms were the center of our approach. However, this one sector approach is not a solution today, so multi-sectoral approaches should be in place to see AMR in the perspective of the ecology or entire environment. This is the concept of the One Health perspective. So we need to understand that AMR from the One Health perspective to map out proper reduction plans. So this is the consensus among research circles. When you look at the right image, so humans are at the center of our approach. So consuming animal meats or considering environment, or we have a lot of companion pets. So we need to take a full holistic approach, not only putting human on the center of this. So One Health approach is utmost importance. And Salmonella and Campylobacter are mainly originated from poultry. So consumption of the meats are ongoing. There are many factors. So when you look at the right image, so the poultry's consumption is going up slightly. So when you look at on your left side, so the broilers are raised at poultry farms, and it goes through the transportation and ending up with table. And we listened from Harim's presentation. He showed us the farming environment, and we had a lot of improvements. So closed farms or houses are our trend. So sanitary gets improved these days in breeding environment. However, foodborne diseases related infections are also on an upward trend. But this story is not only confined to Korea, but also the similar stories can be found in other countries as well. And so please look at the right image. In the US, Salmonella and Campylobacter associated foodborne diseases are mainly attributable to poultry. It's about 20%. And so this is a NPT. So you can see the graph or distribution of non-typhoidal salmonella invasive diseases. So foodborne disease, as you are aware of, are closely related with the sanitary and hygiene environment. So the NTS prone area can be found in less sanitary countries or less hygienic countries. Salmonella and its related diseases are going up. This is not only confined to Korea, the US, and China. And in case of Salmonella in China itself, you can see the distribution of Salmonella enterica cerevisiae in China. So actually, when you look at the distributions here, so there is a Typhonium and Enteritis. So they are major distribution cerevisiae, chicken, pork, beef. So top 15 foods that cause outbreak-associated illnesses So you can find chickens and pork and beef. So there are two main combinations based on data. First one, so retail chickens or duck. So prevalence of salmonella in retail meats in South Korea, and I will talk about Campylobacter genotype and phenotype, and also patient derived salmonella Campylobacter isolates and isolates at a production level from chicken and pork at a farm level. So I would like to compare those data and I'd like to show it to you. And this slide shows the AMR and genotypic profiles of salmonella from poultry meat. So this is about retail poultry meat. So as you can see from this graphic, so salmonella isolates from chicken shows a higher AMR rate compared to the duck isolates. I do believe the reason can be found in the breeding environment. As a disclaimer, this is my assumption. Of course, the chicken meats and the chickens are raised in the improved breeding environment, but this is compact breeding environment for chickens, but for ducks, it is less compacted and less crowded. So this is my assumption as a disclaimer. Anyway, so we look at the salmonella isolates and we look at the taxonomical profiles and chicken isolates, salmonella isolates. So sequence type 32 is prevalent and sequence type 19 for duck is more abundant compared to that of chicken. So ST32 and ST39. So phenotype is infantis and typhimurium. What's interesting here is typhimurium and enteritidis were abundant phenotype, but recently infantis serotype is increasing these days. So Salmonella E. coli strains, what's important here is that ESBL producing strain monitoring. So we look at the Salmonella infantis isolates. So we look at the ESBL producing Salmonella isolates. So we look at the AMR rate, and on your left side, so ESBL producer, non-ESBL producer compared. So there are bigger difference in MDR pattern. So this is well repeated theory in many literature. And on your right side, when we look at the AMR genotype, so ST32, it has CTX-M35 in common. And also, this kind of MDR pattern related genes are identified, similar from the previous slide deck. And now we look at the prevalence of Salmonella in diarrhea patients. So we look at the Salmonella isolates from the feces of the diarrhea patients, and also we look at the layer chicken and broiler chicken and pig. So we do isolation of Salmonella from the manure or feces. In case of human case or patient case, we used fetus in common lab for isolation, and Salmonella serotype varies on diarrhea patients. So you can see a Typhimurium or second one, Typhimurium monophase variant. So these two columns are considered as Typhimurium and Enteritidis and Infantis and others. So you can see multiple serotype distribution from patient isolates. However, when you look at the chicken side here, so Typhimurium monovariant and others for layer chicken. These are the serotype for the layer chicken. When you look at the broiler chicken, actually Infantis is most dominant one. But when you look at the pig isolates, Typhimurium serotypes are mainly identified. So by serotype, we look at the AMR rates. So human-derived salmonella and animal-derived salmonella. We compare AMR rates between the two cohorts. So please look at your right top side. This is a Infantis serotype, and this one is Typhimurium and monophasic variant, and this is Typhimurium itself. So when you look at these three graphs, you can see that patient-derived serotype compared to that, but animal-derived salmonella shows a higher AMR rate compared to that of human isolates. So with these strains, ESBL producer is one of our importance. So we did genotyping for the related genes. So this is for humans. There are many serotypes. So there are various serotypes. So Typhon variant and MTVS, CTSM1 group is the category. But when it comes to Infantis, CTSM9 genes are identified. But for pigs and chickens, when you look at the ESBL producing genes, so those are mostly falling under the category of CTXM9 group. So you can see the similar results found in the other literature. So animal-derived ESBL producers generally are falling under the category of CTXM9. This is a known fact. And hospital-associated, when this is the case, then CTXM1 group was a main one, and our results are in line with those findings, similar findings in other literature. So for clarification, we did a complete genome sequence, comparative genome sequencing. So Illumina platform was used. Then the reference genome should be mapped with ours for genome sequencing. When it comes to this case, the costs are not that high, not that punitive. However, this is not complete genome, so in between there are many noises. So this is one of the shortcoming of this. So for the economic reason, so we opt to use mapping one, but actually for our part, quantitative measurement is important, so we completed with genome sequencing. So Illumina Sholit sequencing, Illumina platform was used, and Longit-based PepBio sequencing platform was used in a hybrid assembly manner. So this is result of that. So with complete genome, we did comparison, and as you can see from this image, so green represents the human-derived strains or isolates, and the purple represents poultry-derived one, and the pink one was derived from or isolated from pigs. When we do comparison, so this purple, this is a sequence type, and with these genomes, we compare genomes with each other. When you look at the sequence type, ST, so MST10 sequence type can be used. However, genes 6 or 7, there are a limited number, so the relevant information was lacking at that time. But genome itself, we use whole genome as I mentioned before. So 3,000 or 4,000 genes were used for comparison, so we can take a deep dive into and we can get more detailed information. But the disadvantage of this is highly costly. Based on the findings, genome sequencing allows for source tracking, so this can be made possible. So in A clade category, human-derived one, and poultry-derived isolates or strains. So they form clade here. So our assumption is like this. So the patients here might consume chicken containing salmonella, which leading them to foodborne disease and CD clad. This kind of patients consume pigs that has salmonella, leading to foodborne disease. So this is our guess or assumption based on our finding. Next one. This is Typhimurium monovariant MVSD. So we did the same analysis. As you can see, for these patients, if the patient falling under category of clade F, then he might or she might consume pigs. And other than that, we are not able to do source tracking, but some other contributors are contributing to the foodborne disease. And this is infantis. So clade B, so chicken originated foodborne disease, and if D clade, then pork associate foodborne disease. And Nomorflora. So salmonella was not a contributor to chicken, but Campylobacteria is one of the contributing factor behind foodborne disease. So we look at the AMR rates of CZ from chickens and duck meat, but there are little difference here, and also from the C. coli from chicken and duck meat. And we did phylogenetic relatedness of Campylobacter from poultry meat, and so when we different sequence type, there are many sequence type. But Campylobacter itself shows multiple sequence types. So from chickens and from duck, the common ST is highlighted in orange. Next, as I saw you from the salmonella case, human-derived isolate or strains that cause diarrhea. We look at the human-derived isolates and also isolates from layered chickens. This is the results. This is the AMR rate profile seen here. Campylobacter, so third generation cephalosporin, fluoroquinolone resistance is very important. So when we look at the genotype, OXA61 genes are abundantly found. 80 to 90% of cases show OXA61 genes, and the other contributor of resistance is dyronase mutation. Dyronase mutation was commonly found. As the comparison of Campylobacter from humans and layered chickens, so I highlighted the sequence type in common. This is clonal compressor, clonal complex 21, 45, 354, 606, 206. This clonal complex associated Campylobacters are commonly isolated, and these findings can be found in a growing number of literature. This is a commonly identifiable sequence type. We did genome sequencing for comparison. So let me consume 1 or 2 minutes more. So when you look at this clade, you can see a duck image. So this pink duck represents pet duck or companion duck, and the yellow one was a straight ducks from the facilities. So you can see the relatedness of genomes. It was linked to chicken meats. I assume that this is relevant with the dog feed. And this is the last page of my presentation. This is the take-home messages and acknowledgements. So we need to monitor AMR bacteria contamination in food distribution stages for risk assessment. And second one, even though it's costly, but we need to apply genome-based analysis for source tracking. Thank you for your listening. So Salmonella and Campylobacter strains are main focus of his presentation from the perspective of Salmonella and Campylobacter, and he also compared and shared with us AMR rates and genome-based analysis was done to see source tracking for humans and for livestock animals. Thank you once again. If you do have questions, I'd like to entertain the question from the floor. Hearing none, I'd like to proceed Q&A session after all presentation ended. Next presenter.
두 번째 연자를
Let me introduce the second speaker, Professor Dominic Haider. He graduated the Minister University in Germany, and his major is computer science, and he also completed PhD, and he studied in the Harvard University and Italy, and currently he is serving as a professor at the University of Münster. And for a long time, he was responsible for the education for the PhD candidates, and he provides various lectures and seminars. The title of today's presentation is AI in Antimicrobial Resistance and Infectious Disease Control. Please start your presentation. The floor is yours.
First of all, I would like to thank for the invitation. It's really a pleasure to be here, and hopefully for the nice introduction. As I'm a computer scientist, my presentation will be slightly different, but I want to show you some of the results from recent projects that we have and ongoing projects from my institute. So I will start with a short introduction, and then I will give you some results from a study that we conducted some years ago about AMR in the environment. And then I want to go over to another project where we used AI and genomics to identify AMR resistance mutations, as well as, oh, there's something missing, about novel AI-based antibiotics. All right, so as you know, AI enters all areas of life. You probably all have an Alexa at home or something like that. And this is also, of course, true for medicine. And with AI, we can basically improve medical treatment, we can improve diagnosis, we can identify novel subtypes, and get insights into the underlying mechanisms. And typically in medicine, these models can use very heterogeneous data ranging from omics data, and of course I will focus on that in my presentation. But just for the sake of completeness, these models could also be applied for imaging data, for clinical health records, and what else. So as you all know, so this is something that you know better than me, these are some numbers from a recent report from the World Health Organization. And the numbers that are important is that they estimate that in 2050 there will be like roughly 10 million deaths because of antimicrobial resistance. And this is something that is super important, and I think it's very important to have a fast diagnosis so that you can treat a patient much earlier with something that works actually. So, but let's get started. So some years ago, and we published that in 2021, we did, in a collaboration with the University of Duisburg-Essen, we did an analysis of freshwater lakes in Europe. So you see here on the left, you can see the different sampling sites, and our colleagues, they did a standardized sampling and standardized sequencing process, and we did the bioinformatics part to analyze the data. And they collected samples from like roughly 300 European lakes all over Europe, as you can see. And it's very important that these were freshwater lakes because a lot of studies have been done in wastewater, but we wanted to analyze freshwater. And we did 16S rRNA sequencing to find out what taxa are present, but also metagenome sequencing to get the AMR genes. And we also did an analysis with the environment around the lake. So we developed a tool that where you can just basically put in the GPS coordinates, and then you get all information that you need from statistics, like the economic value of the region and so on, but also information about farming and etc. And this is a figure from a paper that is not from my institute. It has been published like roughly 10 years ago, where they analyzed wastewater, and we did something similar. So what they did here is they basically showed the different antibiotics here that were developed over time, and resistance profiles here from different sampling sites from wastewater. And you see the older an antibiotic is, the more resistant you will find, which is logical, right? And we tried to do something similar, but in freshwater. Here I just want to highlight some of the drugs that are important and they're used a lot in Germany at least, tetracycline, for example. And here is the result, or one of the results from our study. This is just a snapshot, so it's not all the lakes here because it would not fit one slide, but just a snapshot of the most important things. And here on the y-axis, you see again the different antibiotics, and just to highlight some that I will show you in more detail in the next slides. So this is sulfonamide, this is tetracycline, fluoroquinolones, and cephalosporins. And you can already see that the resistance profiles are very different. Here, as said, you have the different lakes, and the darker the color is, the more genes we find. And now this is put back onto the maps, so you can see for the 4 drugs I highlighted, you can see the pattern, and this differs a lot from state to state and from drug to drug. So you can see that for cephalosporin, there's a lot of resistance in the freshwater lakes. For others, it's much less. So for example, here for sulfonamides, you see that there is much less resistance found. And as said, we also try to find association to parameters such as farming and the surroundings. Actually, we found an association to farming, but it was barely not significant. So it's maybe because it was not enough sampling sites or whatever. So there's a clear trend, but it was not significant as said, so we could not claim this in the paper. Okay, so starting from this, we said, okay, well, of course, it is important to identify AMR genes to get a profile, but of course, I mean, this is how research works, right? People find new resistant genes over time that have some mechanisms. So the idea of this project I want to present now, which was funded by the Ministry of Education and Science in Germany, is to develop de novo models based on AI and whole genome sequencing. So our colleagues from Gießen, they collected a data set of roughly 1,000 samples, whole genome sequences, and phenotypic information from patients from their hospital. We also collected around 1,500 publicly available data sets, which are used later for validation. And we used in our study 4 antibiotics and developed different machine learning models ranging from classical approaches such as support vector machines or random forest, logistic regression, to more sophisticated neural network architectures such as convolutional neural networks. And we did an evaluation, and I would not show this to you if this wouldn't work. So what we got here, and here you can see different metrics, precision and recall, which are used to evaluate such models. You can see that the numbers are quite high for the different drugs, ranging from like 77%, or here it's a bit worse actually, to 95, 90% precision for the different models. And you can also see that for example, the convolutional neural networks are quite good here compared to others, but they all perform quite well, actually. So you can see that from just using the whole genome sequencing as an input and phenotypic information as labeling, you can develop a computational model that can predict resistance for unseen sequences. One important thing to mention, and this is why I showed this on this slide here. So these are the positions that have been identified by the models to be the most important ones for the prediction. However, what you see here is you see the SNP position, the gene region, the corresponding genes, and the corresponding drugs for which they were important. But you can now ask yourself the question, but most of resistant genes are found on plasmids, right? And you don't get them in whole genome sequence, which is actually true. But what we identified here, and the colleagues in the lab are currently trying to figure out what's going on here, is that we think that these are compensatory mutations that are found in the genome, which reflect or shadow the resistant genes in the plasmid. So whenever you pick up a plasmid with a resistant gene, there needs to be some compensation in the genome for the fitness loss, and this is what we see here. So that's our interpretation. The next step in the project was actually, we knew that from other studies that we conducted like roughly 10 years ago on HIV, is that there's a phenomenon called cross-resistance. So whenever you give, in HIV for example, a drug to a patient, at some point HIV develops resistance, and this resistance is not only against the drug that was given to the patient, but also to other unseen drugs. And this is called cross-resistance. And we know that this is also true for antibiotics, and we ask ourselves, so if we know that cross-resistance exists, could we put this information into the computational models to improve their accuracy? And the idea that comes into play here is the so-called multi-label learning. So instead of what I told you before, having an conventional learning where you try to find a mapping from the whole genome sequence to a label, susceptible resistance against drug, whatever. You now have multiple labels that you want to model simultaneously. And with that, you can model the interdependencies between the labels into the model and hopefully improve it. And there are many, many approaches. I don't want to go into detail here how to model that, ranging from like very simple ones like classified chains to more sophisticated ones. And you can also basically use any kind of base model for that. But I just want to show you here the results. So here you can see the different colors are different approaches for multi-label learning. But what is important on the slide is what you can see is that by using the multi-label learning and the information about the dependencies between the drugs, the prediction can be significantly improved, as you can see here, which is in particular for the model ECC, which has been shown in many studies to be the best way of doing multi-label classification. So this is also true here. We get significantly better results. For example, you can see here, this is much better than the other models. Okay, so as you know, there are many, many different antibiotics available, but we restricted our analysis to just 4 of them. And the reason is that for some of the antibiotics, we simply had not enough data. So for example, for some, there were just a few resistant samples, and for others, there were just a few susceptible ones, and so on and so forth. So still, we said, okay, we have the data, and if we don't use it, we waste it, right? So is there a way to also develop models where the number of samples that you have for training is very low? And fortunately, the answer is yes. And I want to give you a very easy real-world example which explains how this works. So who of you have learned to play an instrument? Don't be shy, raise your hand. Yeah, so let's imagine you started as a little girl or little boy and your parents forced you to learn piano, right? So you not only learned to play the piano, but you also learned to read a melody from paper, you learned what rhythm is, and so on. So you learned all the stuff around that, right? And now let's imagine years later, you decided to learn another instrument, let's say the violin. So again, you have to learn how to do that, right? Because it's a completely different instrument. But you don't have to learn these two again. You don't have to learn what melody is, how to read it from paper. You don't have to learn what rhythm is. So you don't have to learn from scratch. And this is the analogy here. So there are models, and these are called transfer learning, where you basically train a first model on a data set where you have a lot of data, and then you use this model and fine-tune it with a smaller data set and develops with that a second model. So let's say for drug A, we have a lot of data, we train the model, and for drug B, we just have a few samples. So we take this model here, fine-tune it with the small data set, and then it works. Because the model not only learns the prediction, it also learns how whole genome sequences look like, you know, what the structured SNPs are, and so on and so forth. And here are the results published some years ago. Again, we used the 4 drugs here as an example. We trained the model, and then we transferred the model to the first model where we had the most data. We transferred it to the other drugs and did a validation on the publicly available data set as well to show that it's not overtrained to our data set. And this is the result. So you can see here in blue the original model, in yellow the one after transfer learning. So you see a significant improvement on our own data, but also on the public data that was not used at all for the model. The metric that is used here is MCC, which is one that is very common in computer science, but not so much in medicine. It has some advantages. I don't want to go into detail here. Just if you ask yourself the question what MCC is, it's a different metric. The higher, the better, as always. Okay, so this is the first 2 projects, and the last projects I want to show you is one that we have recently submitted for funding at the ministry, and fingers crossed that it will be funded. The idea behind that, and this is a figure taken not from a publication from us but from others, that we want to use AI to develop novel antibiotics. So the problem with antibiotics is that developing new ones is scientifically and economically challenging because they are very cheap. So investing a lot in the development of new antibiotics is highly costly, but the revenue is low, right? And antimicrobial peptides could be a promising alternative because they have some very good characteristics, and there are some examples already from nature, for example, which show that they work and could be a good alternative. So there are different mechanisms. They can disrupt the membrane, but they can also interfere with proteins from the pathogen. So there are many, many different ways of how they could act as an antimicrobial drug. So what we want to do, and this is a paper that is currently under review, we developed a database which is currently the largest one for AMPs, our curated database. It has like roughly 75,000 sequences with all information that was available, and we developed a model that is based on the GPT architecture. So this is the same architecture as ChatGPT, for example, but it was developed specifically for protein sequences, so not for text, right? And so the original model was ProGPT for protein sequence, which was not developed by us but by others, and we developed a version explicitly for AMPs, AMP GPT-2, which was trained on the compass data there, and it can be used to generate new AMP sequences. And we also used a prediction model from the literature to find out whether the predicted AMPs or the generated AMPs are actually AMPs and not just peptides, right? And the model from the literature says that more than 95% of the generated sequence are actually AMPs. And then, of course, we wanted to test that in the lab, whether this is true. Here is just, here's a plot where you can see the generated sequences. So all the dots are generated sequences, basically. And here you can see the distribution on a UMap for the data from Compass, from ProGPT, and from our AMP GPT. And you see that the generated sequences better reflect Compass, which is obvious because they were trained on, but you can see that it's very different to the original ProGPT model. And as said, we tested it in the lab. So we tested 2 AMPs that were generated, and one was working, the other one was not. So 50-50, it's not 95%, but still we found a new AMP, this AMP 28 here, we can see that it significantly reduces the growth rate of Klebsiella. So it is working in principle. And what we now currently want to do in the proposal I mentioned before is we want to further improve the AMP GPT models by introducing so-called LoRa's, which are adapters that can model specific characteristics that they want to optimize in the peptides, for example, solubility, toxicity, and so on. So there are different ways. And with that, you don't only get potential AMPs, but also ones that are soluble and not toxic for human cells. And with that, I want to thank you for your attention, and of course the people in my institute, because they actually do the work. And so thank you very much.
네, 감사합니다.
Thank you very much. The speaker talked about the development of new prediction model to see the relations between the antimicrobial resistance and And secondly, he also talked about genome sequencing using AI and antimicrobial peptide-based antimicrobial products development was mentioned as well. Thank you very much.
The third presenter is from Dr. Metulamudir, and he received molecular microorganism in Coventry University for 7 years. He carried out research in the field, mainly RNA, DNA, protein isolation, and construction recombination DNA are the focus of his expertise. And as of now, he put a focus on biological standardization, and MHRA innovative research are led by Dr. And also he made a lot of publications of high reputation, and he also carried out research on AMR. And the topic of his presentation is building trust in phage therapy, reference reagents for safety and quality assurance. Doctor, the floor is yours.
Thank you very much for the invitation to speak here today. And thank you for the introduction. So I am Matthew Lemordier. I'm a senior scientist at the Medicines and Healthcare Products Regulatory Agency, which is the equivalent of the MFDS in the UK. So we're the UK's medicines regulator. We regulate medicines, medicinal products, diagnostics, vaccines. And essentially, we play the same role as the MFDS, so we make sure that these products meet standards set for quality, safety, and efficacy. Primarily, safety and quality is the focus of the MHRA's functions. So I have a slightly unique role within the MHRA. So the MHRA has many different responsibilities. These are the ones I will focus on in my work, and they evolve around ensuring the standard of products produced and entering the UK market for medicines, primarily for human use, but I'll go into it a little more detail later on in the talk about how our work spans the One Health space. We make sure that the products meet these criteria, and also we do collaborative research at the science campus at the MHRA to enable innovation, and particularly innovation in the space of antimicrobial resistance. So I'm part of a very small team at the MHRA. There are only 3 of us in my team, and we are funded by the UK government's Department for Health and Social Care, and they have the Global Antimicrobial Innovation Fund, which supports our work. And the remit of our work is to tackle antimicrobial resistance through supporting innovation with regulatory science, with a very strong focus on low and middle income countries where the greatest burden lies, and across the One Health space. So we are working with global regulatory authorities as well as cross-agency within the UK government, working with Veterinary Medicines Directorate, Food Standards Agency, the Environment Agency as well. So the GRAMRIF program was set up in 2023 and funded the MHRA to provide regulatory support to innovator accelerators which receive GAMRIF funding. These innovator accelerators support projects across the One Health space in food, veterinary, and human. As you all know, the financial incentives behind developing novel antimicrobials are not very strong, and therefore the innovation burden is falling to small groups of academics and small and medium-sized enterprise. This is very challenging for them because large pharmaceutical companies have big legal divisions which are constantly in contact with the regulatory authorities, and they have a very good understanding of the requirements for quality of data that they need to produce in order to progress, the way they need to design their clinical trials to satisfy the regulatory criteria. But small groups of academics and the SMEs, they're very much focused on the science, they're not regulatory experts. So GAMRIF saw this need for regulatory support in the AMR space and therefore provided the MHRA funding to provide this support to these innovators. One of the things we found very early on was that bacteriophage, so these viruses that can target and destroy bacteria, of course, a very promising alternative antibiotic. There is great promise for these products moving forward to tackle antimicrobial resistance, and I've just laid out a few examples here why they show so much promise in the space. So they are extremely they have high specificity when they're targeting bacteria. They have very little or no collateral effect on commensal bacteria, so they won't disrupt the larger microbiome of a patient. They also display synergistic effects with antibiotics. So if we see a multi-drug resistant bacteria has colonized the patient, using bacteriophage in combination with antibiotics can resensitize those bacteria by adding additional fitness burden to them, in addition with those traditional antibiotics. They've also shown strong antibiofilm activity, in vitro particularly. They can break down those biofilms, disrupt them, and allow the penetration of traditional antibiotics to take effect. As bacteria and bacteriophage have evolved over time, there is a constant arms race happening between the two sides. The bacteria develop resistance to bacteriophage, but the rates of mutation in bacteriophage equal that of bacteria, so they're able to compete and evolve at the same rate to develop new mechanisms of killing bacteria. So while antibiotics take a long time to develop, natural phage in the environment are evolving on their own to be able to fight these new waves of bacteria we're seeing. And also, we can customize these bacteriophage to change their target or even to modify them in a way that makes their manufacturing process more efficient, which is going to, of course, increase return on investment for those manufacturers. Bacteriophage have been used in the food industry, at least in the USA, since 2006. They're regulated by the FDA under the GRAS status, so generally recognized as safe. There are a vast number of bacteriophage products regulated under this GRAS status by the FDA, and I've just listed here some examples of bacteria which those phage products target and the types of food products that they are being used for. This is both monophage, so single phage products, and also cocktail phage products, sometimes up to 9 or 10 different bacteriophage being used. They're also used in the UK. Examples I know of are in bagged leafy salad, in cheese production to prevent the growth of listeria on the outside of the cheese, and to prevent potato rot during storage of potatoes. So they are widely being used in the food industry at the moment, but in human use, there are a few challenges to employing them in healthcare services. They have been used for over a century now, particularly in Eastern Europe. However, when Fleming discovered penicillin and the advent of what we call now traditional antibiotics, this took over and bacteriophage took the back seat. However, now we know that bacteriophage pose such a promise to tackle antimicrobial resistance which has emerged. It's got everyone trying to incorporate bacteriophage into clinics, especially across Europe. This poses a problem because regulatory authorities naturally evolve over time to deal with the types of products which are in common use, and it's the role of the regulator to try and evolve with novel therapeutics coming through the pipeline. And at the moment, that seems to be a big drive behind bacteriophage. However, there is a consistent inconsistency in the quality of clinical use data, because bacteriophage have only ever been used for compassionate use in the West. And therefore, the reporting and the quality of this data is very varied, and it's hard to build up a clinical use case. And most clinical trials have failed after phase 1. So they have very high efficacy in vitro, but in vivo it's much more difficult to prove clinical efficacy, and the reporting is not standardized. Regulatory clarification is needed. As I said, national regulatory authorities evolve over time, and the frameworks are built to accommodate the medicines in use. Novel products come and they say, where do we fit in the framework, the regulatory framework? We think we fit here, but some aspects of our products are not covered by the regulations which exist. So there is a need for clarification around the regulatory strategy to deal with novel therapeutics such as bacteriophage. The biggest problem lies in engineered and synthetic bacteriophage rather than natural bacteriophage. The need to clarify what good manufacturing process looks like for the manufacture of bacteriophage is sorely needed. And there's also, as I said before, uncertainty around the reimbursement model, how manufacturers and developers going to get a return on investment on this type of product when the market forces are as weak as they are. And lastly, what I will cover next is the lack of appropriate reference materials, which is a term not many people are familiar with. Product developers and manufacturers, yes, but as I said, academics and SMEs maybe not so much if they're not up to speed with the regulatory jargon. I think a lot of people are familiar with the term standards, but it's usually written standards, and I will be talking about physical reference materials. So the UK has come a long way in terms of clarifying the regulatory space around bacteriophage products. We recently, in the last couple of months, released a regulatory considerations document to show innovators that there is a space within the framework for natural phage products as a biological medicine. The framework exists, and this document shows the way in which bacteriophage products fit. It signposts parties to the relevant regulatory requirements and the need for evidence generation to support clinical trials, the manufacture and the import of investigational medicinal products, so for clinical trials use and for compassionate use, and the need for compliance with good manufacturing processes. And I've linked it here for anyone to look at that document after the fact. The European Commission's EDQM has also published a general chapter on the manufacture of phage products, which has also gone a long way in clarifying for product developers the need to fit within existing regulations for phage product development. So physical standards, what are they and why do we need them? So researchers, developers, manufacturers are required to use physical reference material. You can think of them as a control or a calibrant, essentially to validate a bioassay for biological assays or physiochemical testing that they do during the production of a product. And it tells the regulator that they are assured of the quality of the data they are producing, the methods they are using are sufficient to produce high quality data, and it's reproducible. And regulators need to see that. I know we spoke about the VICH yesterday, but the ICH, the equivalent for human medicine, specifies the need for these reference materials in product manufacture. And the MHRA produces over 95% of all WHO international standards, so the physical reference materials used across the world. So this is our bread and butter at the science campus of the MHRA. So what do we need reference materials for when we're talking in the context of bacteriophage? Or really, this example applies to all novel therapeutics, the need for reference materials, but I focus on bacteriophage here, because this is my role within the MHRA. We got funding a couple of years ago from the Food Safety Research Network to produce an initial trial reference material. This reference material was a metagenomic cocktail of bacteriophages. And this was purely to validate library preparation, sequencing, and bioinformatics of bacteriophage products, to confirm taxonomic identification and the presence or lack thereof of deleterious genetic elements. So AMR-associated genes, toxin-encoding genes, integrase genes, for uses in biobanking, tracking genetic drift of bacteriophage products over time, and also surveillance, among many other applications. This was just the beginning of the work on bacteriophage reference material, and it has evolved following the funding through GAMRIF. We're now trying to develop a material which has much broader use rather than just to validate sequencing approaches. So while sequencing is still a cornerstone of validation for bacteriophage products, we're also conducting a study in collaboration with a bacteriophage manufacturer on optimizing the freeze-drying process, lyophilization, looking at how different formulations affect the stability of freeze-dried bacteriophage over time, how much can we maintain viability with different formulations and different titer of bacteriophage, either monophage or cocktail products. We're also looking at a reference method in mass spectrometry for the verification of the absence of host cell proteins, which is a requirement for all biological products. They cannot include host cell proteins within the final product. And for bacteriophage, this is key because they are, of course, fermented bacterial products inside these large cultures of bacteria. We need to ensure that there are no toxins and no host cell proteins being carried forward during the manufacturing process. And also integrity of the bacteriophage. So what does that final product look like? Of course, under the microscope, you can see you have so many bacteriophage, but can you validate that their capsid actually contains a viable genome? Or is the tail of that bacteriophage broken? So how much of what you know about your phage titer is bound in reality? Much like with bacteria, we use colony-forming units. With phage, we use plaque-forming units. But this should, after the manufacturing process, be validated through the use of integrity assessment. How many of those capsids actually contain a viable genome? And that will help with dosing regimens and ensuring that patients are receiving a dose that the clinician thinks is actually present within that product. And this underlines the work that will underpin a collaborative study moving forward. When the MHRA develops these reference materials, we develop them in collaboration with product developers and manufacturers who often donate material for us to work with in order to develop these reference materials. At the moment, I am in an information gathering stage after the initial trial, and I'm openly looking for collaboration with the development of these reference materials. So I openly ask the audience, anyone who is either involved in bacteriophage, either directly or indirectly works with colleagues who are working in bacteriophage space, please contact me. I would love to tell you how you can input into the work that the MHRA is doing. At the moment, I'm looking for input to guide the design of this reference material, which will be used by product developers across the world, hopefully with the scope of becoming an international standard endorsed by the World Health Organization. So I know I'm a bit short, but thank you very much for listening, and 감사합니다.
Thank you, Doctor, for your thought-provoking presentation on bacteriophage control on AMR and the bacteriophage application in the food industry in the USA and the UK. And naturophage as a biological agent, related regulatory documents were written, and validation requires reference reagents for bacteriophage, and there are many R&D efforts ongoing. Thank you.
집중하다 보니까 벌써
And already it's the last presentation, and let me introduce the speaker. The fourth speaker is Dr. Sanyoung Sonu. She received a PhD in the veterinary pathology at the College of Veterinary Medicine at Konkuk University, and she is an expert in disease of poultry and pigs, and she also works with the government agencies and associations, and she also provides advisory services to many companies. For the last 27 years, she has many experiences contributing to the academia and industry as well as the government, and she is currently the head of the Green Cross Veterinary Bio Research Center. Please go ahead, and floor is yours. Hello everyone. It's my pleasure to meet all of you. And my presentation is not academic. I would like to give you the overview. So I would like to make a presentation in Korean. I hope that you can understand it. As explained by the chair, I'm sorry, I'm quite short, so I will move forward. For the last almost 30 years after majoring the veterinarian pathology, I have been engaged in vaccine development for a long time. And I also participated in the field as a practitioner. And this is the title. And I also reviewed what I studied, and I also look back at my experience at the field. In the beginning, of course, I will cover this in my slide. In 2005 and 2006, the antibiotics have been regulated, and across the world, the regulation has been strengthened from 2010 onwards. But even before that, I also was active in the field. We used antibiotics a lot, in particular in the farm. But now in Korea, the regulation is very strict. So I believe it is well controlled and organized. In regards to this, I would like to start my presentation. Today, I would like to focus on the reduction of antibiotics use, in particular in the livestock field, what kind of efforts have been made, and what we need to do in the future. This is the order of my presentation. Yesterday, many presenters covered this. There is a site called Animuse, and there is annual reports. And I extracted 2024 reports. There are many countries as a member of OA, and this is the reports from more than 150 countries. And this is the annual report. Around 74,000 tons of antimicrobials are used. And in Korea, we also use a lot of antimicrobials. And when you look at the global market, the entire animal health market is indicated as $5.22 billion US dollars, according to this report. And I believe the total size of the animal health is $660 million US dollars. And Korean account for 179 million US dollars. And overall, we can see the upward trend. When it comes to antimicrobial, tetracycline is number one for a long time, so it is widely used, followed by penicillin and macrolide. And we also need to look at this veterinary clinically important microbial. For example, fluoroquinolones and polymyxins and the third and fourth generation of cephalosporins. These are widely discussed. As for the fluoroquinolone, there is enloxacin. Enloxacin has been used for animal for a long time, and the efficacy is very strong, and it is even used nowadays. And there are agents based on cephalosporin, such as cephalosporin. It is used for the respiratory diseases, and the usage is increasing nowadays. So the later generations of antimicrobials are used more nowadays for animal. And how can we carve down this usage? That is important part in antimicrobial usage. As for the AMU, we need to also look at the aquatic food producing animals. This is quite critical, even though the regulation is not that strong yet. So we need to have cautious approach. And another part we need to look at is the antimicrobials in companion animals. And I also wanted to point out this, how much has been used in which region. In Europe, there are many countries, but the usage is only a tenth of Asia Pacific region. So in Asia, we use a lot of antimicrobials. It is obvious because there are many complexes producing animal, and as for swine industry, it is concentrated in the Asian region, and poultry industry is also concentrated in Asia. So that is why there are many usage of antimicrobials. So in Asian region, how can we control and regulate them is important. And I also marked stars. As you can see, tetracycline is number 1, and as for fluoroquinolones, is used for aquatic animals, which is indicated in blue, but we are not that focusing on this one. And for the terrestrial animals, tetracycline is used, and cephalosporin is used for companion animals. And even though the figure is not that great, it is also used for the terrestrial animals. Moving on. Yesterday, there was a presentation from a professor. When we look at the antimicrobial usage in biomass, it is decreasing. The figure is 97 milligram per kilogram, but in Asia Pacific region or Africa, basically the usage is decreasing in Europe and Africa, but in Asia Pacific region, we can see the usage is still very high. And 21 milligram per kilogram is indicated in the aquatic food producing animals.
실제 사용하는 건 이런.
Let me sum up. So we looked at the usage. Then what about the regulations? First of all, in Europe, Korea, USA, and Australia, the regulation is very strict. In these regions, food safety and hygiene are highly focused. So this is a high-class standards. But there are Southeast Asian region and Africa which are not regulating these antimicrobials because they sometimes just recommend it, not mandatorily regulate them. And we also need to see the statutory regulations. In Korea, it is well established, and prescription system is also important. Yesterday, there was a presentation from Denmark. In Europe, there is a veterinary prescription for all antimicrobials. In Korea, from 2013, the system has been introduced, and in the US, it was introduced in 2011 and 2017. And in Australia, it is the same with the EU. The regulation is very strong. And let's take a look at the usage of growth promoters. Without these regulations, there were a lot of use of growth promoters. Even though the animals are not sick, in order to increase the productivity, they add it in the feed. sometimes we were able to see the feed with antimicrobials. But it was totally banned, and there were many backfire from the farms and the industry. But in 2006, Europe introduced this ban on growth promoters, and Korea also started banning this growth promoter. There were many conflicts and confusion, but now it is stabilized. It is the same with the US from 2017, and prophylactic use. Sometimes it is principally prohibited, and in some countries it is under management, which means there must be control of the veterinarians and consultation. I believe it is quite important. In the US, the prophylactic usage is allowed only when there is a veterinarian authority. And the vets need to control because they are the experts. So in all these countries, they are talking about that. And there is a surveillance system. In Korea, we have a very well-established system. One thing I would like to mention is HCCP at the slaughterhouse. From the early 2000s, we started this program, and I participated in the HCCP consulting for the pig farms. And at the time, the Ministry of Agriculture had very strong stance. So for the pigs, bovine, and chicken, they drive this program. They regulated the introduction and the usage of these drugs. And here, in the meat, they saw whether there is any residuals, and they wanted to control the appropriate level of usage of antimicrobials. So this is the overall situation in many countries. And today I would like to focus on the vaccine. I've been studying vaccine until now, so I would like to cover this part. In order to reduce the usage of antimicrobials, there are many alternatives, as you can see here. Probiotics, prebiotics, and herbs and immune stimulants can be used as a supplement. But as indicated in yellow color, vaccine is the most effective measure. When we use appropriate vaccines, we can appropriately prevent the diseases, and we can also reduce the use of antimicrobials. Yesterday, the presenter from Harim said this: biosecurity is very important, and we also need to control the environment to prevent diseases and reduce the use of antimicrobials. And without regulation, actually it is difficult to execute. So regulations and policies are also critical. Let's talk about vaccine. As for veterinary vaccine, it has been studied for a long time, and it has been developed for a long time. So it is the most effective way. Then with the use of vaccine, how can we reduce the use of antimicrobials? The onset of the disease can be decreased or prevented. Then we can actually reduce the use of antimicrobials, and the spread of AMR can be also suppressed, so that we can achieve animal welfare, food safety, and public health. This is kind of virtuous cycle. So vaccine plays very important role here. Then, what is the effective vaccine? If the vaccine is effective, then there must be economic advantages. For example, in treatment, we use a lot of antimicrobials. With the vaccine, we can save the cost for treatment. We can also reduce the management cost and improve productivity. Secondly, we can also increase social trust. In many countries, they have the strategy for vaccination. Let me talk about the global market situation. The total market size is $6.33 billion US dollars, which is bigger than antimicrobial market. In Korea, the market size is 300 million US dollars, which is quite big. And as for veterinary vaccines, inevitably there have been continuous development because disease have mutation continuously. As a result, inevitably development should be continued, and there are regional variations. One vaccine can be effective in one region, but it is not effective in other countries. And there are emerging infectious diseases. It can be caused by virus or bacteria. And when there is infectious diseases, to carve down the symptoms, we first use antimicrobials. So, vaccine must be developed for emerging infectious diseases. And we also need to think about more effective and less stressful administrative routes. Vaccines have been developed as an injection, but it can be stressful for animal, and now we can sprinkle the vaccines, or we can orally administer the drugs, or we can use biology as well, and we are developing new delivery systems as well. With a small antigen, we can extend the efficacy. And usually the injection is via IM, but we can change that to intradermal system. So we can see the development in the sector, and efficacy and safety should be enhanced. When I develop vaccines, these are the challenges, and actually these are the things I need to address. We need to develop effective vaccines so that we can prevent many diseases, and we can also reduce the use of antimicrobials. So it goes on and on. Let's take a look at veterinary commercial vaccines. We can classify them into bacterial infection vaccine and viral infection vaccines. As for antimicrobials, you may think that we can just focus on bacterial infection vaccines. I studied pathology, and when we dissect the carcass, the main source of the disease is viral disease. However, the secondary cause is bacteria. So, for the farms, antimicrobials are inevitably the solutions. When we develop viral vaccines, you may think that it is not relevant to the use of antimicrobials. However, it is very closely related because it can protect the secondary infection of the bacteria. In particular, when it comes to the bacteria infection vaccine, it should be preventive, and it should be very specific. And as for viral infection, it is a systemic disease. And there are diseases suppressing immunity, such as AIDS for human being. PRS for pigs can be the example. And pneumovirus infection can be another example for the poultry. If there is a viral infection suppressing the immunity, obviously we can see the increase of the infection from bacteria. So when we look at most of the veterinary vaccines, we are trying to prevent a viral infection, and that is the main market. So that is a way to reduce the use of antimicrobials. And for terrestrial animal vaccine, it is well organized. For example, poultry, chicken, and swine, they are in herd. In a small space, there are a lot of animals. So inevitably, we see a lot of diseases. And to prevent the onset of the disease, a vaccine is needed. So that sector is well organized. If you have any chance, please look at the vaccines for the chicken and swine. Then you will understand that it is well organized, even better than the human vaccines. And as I mentioned before, as for aquatic animal vaccine, the importance is less put. Why? Because not in many countries there is aquatic culture, but in Korea we have aquatic culture. In Korea, we have a number of vaccines authorized by the government for aquatic animals. That is why I said not well organized regulation and compliance. We need more interest and attention to this area. And as for research and development, we need to look at various factors. I wanted to emphasize aquatic food producing animal vaccine. In Korea, we have 29 authorized vaccines for 10 pathogens. In Southeast Asia, the market is growing exponentially. That is why we can see a lot of development for vaccines, and vaccine use is also increasing. Chile, North America, and Europe, vaccine is also used. As for market share, bovine accounts for lion's shares because it is the biggest usage. Currently, I'm part of Green Cross Veterinary Vaccine company. So this company has the longest history in Korea. We have 52 years of history. Then at Green Cross, what are we doing? I believe I can cover all the fields we are active. We have more than 102 approved vaccines, and some of them is currently used, and some are not used anymore. And we have vaccines for swine, bovine, poultry, and aquatic animals. We provide a total service. So, of course, we sell antibiotics and pesticides. And when you look at our portfolio, it is very wide. Our main business is vaccine development. These are the vaccines we currently develop. There are bacteria-based vaccines and viral-based vaccines. So viral infection will eventually reduce the use of antimicrobials. So viral vaccine development is also critical, as I mentioned before. We have poultry vaccine, canine vaccine, and aquatic animal vaccine. Aquatic animal vaccine has been for the first time approved by GC, and it is mostly applied to turbot. And currently, we are looking at shrimps and other species. Let me give you the conclusion. This is kind of summarization of what I talked. And this is the future steps. First of all, as for vaccine, it is the most effective way to reduce the AMR. So, there must be more research and development in the future. Then what should be the focal point of R&D? Multi-pathogen targeting vaccine development? Next generation vaccines? We are currently developing it actively, and we are looking at mRNA, DNA, vector vaccines, etc. And economic assessment should be conducted, and we also need to look at One Health related research. And another point I would like to make is vaccine efficacy and economic assessment should be done. Furthermore, then we can emphasize the use of vaccine in the future, and education and consulting is also critical. How can we use the vaccines? Should be educated. We have many regulations and rules. Already, we have a lot of regulations and consensus made by many agencies. But what is important is execution and how to. Thank you very much. This ends my presentation. Thank you very much. She talked about the regulatory status of each nation and the importance of surveillance system and to reduce the AMR. The accurate usage of vaccine is important, and currently she also introduced the current area of development. Then from now on, we would like to start the Q&A session. We are behind the schedule, but still we will start the Q&A session. So please move forward. We would like to invite all the speakers to the podium.
자유롭게 질문 있으시면
Feel free to raise your hand if you have any questions. Hello everyone, my name is Jo-mi from Codex. I have a question to Professor Tae-jong-chan. You studied the commercial chickens, and I have a follow-up question. In Salmonella, ciprofloxacin, antimicrobials 100%. I think I saw that in the presentation slides. So what is the meaning of 100%? For example, you studied 100 cases, and are you saying that all of them found the microbials, or all the Salmonella showed resistance? If that is the case, then what is the percentage of resistance in the sample? Because people can misunderstand, thinking that all the chickens commercially available have ciprofloxacin. So please clarify that.
Thank you for your question. So 100% AMR rate to ciprofloxacin, but at the AMR resistance in the slide deck, there are 100 samples, and out of 100, 10 show the Salmonella, and its then positive rate is 10% in ESBL producing salmonella is 1, then it's about 1% out of entire 1. That was our calculation method. So 100% of AMR, then ESML producing isolate, then it showed resistance to ciprofloxacin. So this is the right interpretation of my presentation.
To the Dr. Sonu. Actually, I'm from the Jeonnam National University. I'm the Poki Jones. I got a few questions about the vaccine. Actually, my study focused on the goat, but unfortunately, we do not have any vaccines for the goat in the South Korea. Do you guys have some plan for to develop the vaccine for the goat industry? If you have, which one is the first?
네, 염소에 대한
Well, as for the goat, I believe it is an emerging issue, and there is no approved vaccine for the goat. So many researchers are trying to develop the vaccines for the goat. We are taking the first steps. Goat is similar with the bovine. They have the ingestive diseases, so that should be the first focus in vaccine development. And another topic is tuberculosis. So I believe the development is focusing on the digestion and TB, and another one is a foot-mouth disease. And I believe there are many researchers, and there will be more vaccines. Actually, we have one in the portfolio.
Thank you to all the speakers. Very interesting topics. My name is Myung-Shin Che from Codex Secretariat. I have one question to Dominic and one question to Matthew. First question goes to Dominic. AI is always interesting and potential area. You are developing AI prediction models. How do you validate models, and what will be the impact on the development of AI tool on the real research area? And another question goes to Matthew. It's very interesting to hear about microphage product. Are there any product already in the market? And reference material is very important. How do you ensure the stability? Because mutation is the issue. And perhaps the manufacturer need reference materials. In that case, do you develop secondary reference materials? In that case, how do you validate? Thank you.
All right, thank you for the question. So typically when you develop AI models, you do internally a so-called cross-validation with your own data, but this is not enough. So what you additionally have to do is to have a second cohort of data that was not used in the method development or the model development at all. And then you can evaluate how the model behaves on new unseen data. So that's the general idea. So you always need a validation cohort. However, with that said, we are completely in the field of research only. So whenever you want to go into translation and really use that for making medical decisions on patients, then you have to go all the way to regulation. And with that, I can hand over to my colleague.
Thanks for your question. They're really good questions. In terms of phage on the market, there are no approved medicinal phage products on the UK market, apart from those used in the food industry. In terms of stability, we will do long-term stability studies on all reference materials that we develop. It's part of the criteria for development. These include, many of the reference materials are lyophilized, freeze-dried, to ensure stability, and also cold chain independent so that they can reach as far across the globe as possible. So those studies are undertaken to make sure they're as stable as possible, and the shelf life of those products is always communicated in the instructions for use of those products. And in terms of secondary reference materials, we do develop across the spectrum of reference materials, from primary calibrants as international standards, and then secondary calibrants that will be calibrated against those initial international standards. So that's how we would validate those secondary reference materials. Thanks.
네, 시간
So we have on our schedule. So you can approach to your speakers after the session. There are 2 online questions. So please return your answer. The first question goes to Professor Chejong Chan. It's written in English.
Low middle income countries regarding setup of WGS lab, or how to start up with the WGS testing of their AMR surveillance isolated to generate data.
지놈 시퀀싱 데이터와
Genome sequencing data related question. So when we receive isolates, then with that strains, we go for the taxonomical identification, and also we identify phenotypes of AMR. After that, genome sequencing follows. Then, as I mentioned in my presentation, Illumina platform, PetBio platform are co-used to earn hybrid assembly process based on that data generated. So when we go for the assembly itself, Unicyclona, Kano, 2 programs are used for our case. So after assembly being done, database, we utilize it for comparison to go for the genome sequencing and AMR determinants to be identified. And this is the process. And when it comes to the comparison itself, so our sequencing results and similar genomes on database are compared. So this is how we proceed. I hope this clarifies your question.
조금 어려운 질문. 기반 구축이나.
It is a quite difficult question, and how can we start up the infrastructure? I believe it is quite complex to answer. And second question, thank you very much for the presentation. I have a question to Dr. Sonyo. You are studying aquatic vaccine, and is it the vaccine for the actual fish? And I believe it will be used in the aquatic culture farm. And most of the farms have large scale and many species. So how do you inject the vaccine? For now, this aquatic vaccine is widely used. How can we apply this vaccine to the aquatic animal? So we can merge them into the vaccine, or we can inject the vaccine. And it can be cumbersome, but still many farmers use that methodology. We are running out of the time, so we'll wrap up here. Thank you very much for all speakers and participants. And this ends the session 3. Thank you very much.
So the session 3 of the day 2 completed. Once again, please give our chair and our presenters with a big round of applause. Thank you. And now we're about to commence the session 4, but before that we do have a 35-minute long break. So in the front of conference hall A, there are e-posters established, so you can scan your QR code on a flyer to see the presentation slide decks, and session 4 will begin at 20:45. So I will be back soon.
At 12:25 again. Thank you. antimicrobial drugs or antimicrobials to treat infections caused by microbes, including bacteria, viruses, fungi, and parasites. But what if these microbes started fighting back and the drugs stopped working? You may not have heard about it, but this is already happening. The microbes are resisting. This is called antimicrobial resistance, or AMR. It means that in some cases, common infections can no longer be cured. This is a serious threat to human health. But now that you know, you can help do something about it. AMR happens when microbes are exposed to antimicrobials too often or in the wrong way. As a result of this, some microbes survive because they have developed a defense mechanism to resist the effect of the drugs. These stronger microbes can grow and multiply in the presence of drugs, passing their resistance through their DNA to future generations. Over time, the resistant population increases and the antimicrobials that once killed them no longer work. Microbes can be present in the food we eat, and some of them can make people sick. These are called pathogens. These pathogens can also contain genetic material that enables them to resist antimicrobials. So if we eat food containing AMR pathogens and get sick, the illness can be more difficult to treat. The transmission of AMR microbes to people through food is called foodborne AMR. The overuse and misuse of antimicrobials in food production, along with poor hygiene and improper food handling, contribute to the burden of foodborne AMR. If you grow, produce, process, sell, or eat food, then you have a role to play. That means there is something we can all do to tackle foodborne AMR, whether we are farmers, veterinarians, food producers, or consumers. We can help slow the development of AMR by using antimicrobials responsibly, which means only when needed and as directed by a relevant health professional. We can help prevent the spread and growth of AMR microbes by using good hygiene practices and safe food handling and storage practices. Additionally, thorough cooking can kill pathogens. And if you are a government policymaker or a manager in a food business, you can use the Codex Alimentarius texts on antimicrobial resistance. These provide guidance on how to minimize and contain AMR-related risks along the food chain.
All about Codex standards on AMR and its implementation.
Codex standards are the benchmark standards for food safety under the World Trade Organization's agreement on the application of sanitary and phytosanitary measures. They are science-based and agreed upon by the 189 members of the Codex Alimentarius Commission. Texts are developed within specialized Codex committees.
We had a task force of AMR, Task Force Antimicrobial Resistance, hosted by the Republic of Korea. We did develop the guidelines for integrated monitoring and surveillance of foodborne AMR and the code of practice to minimize and control foodborne AMR.
In addition, Codex had developed the guidelines for risk analysis of foodborne antimicrobial resistance.
We were privileged enough to have the funding from the Republic of Korea to take these documents out of the printer and go to 6 countries and implement it.
The ACT project is supporting the implementation of Codex AMR texts in the plurinational state of Bolivia, Cambodia, Colombia, Mongolia, Nepal, and Pakistan. The support is framed around 3 interconnected pillars: improved surveillance, governance, and awareness of AMR.
When we started that project, we learned that the situation of countries and their needs were different.
Our approach was always tailor-made, and after we identified the gaps, then we developed the activities plan and implemented them, translating what is written in the standards to actual actions at the country level. And I think this was quite unique. Overall, the 6 countries, I think we achieved major success regarding all these 3 pillars of the project.
Actions now will take time to impact on AMR containment. But monitoring and surveillance, policy action, and improved awareness do lead to impactful, collective, and positive results.
The goal is to reduce antimicrobial use, that we control antimicrobial resistance.
El proyecto ACT está ayudando a los países a implementar las directrices del Codex en la vigilancia y monitoreo de la resistencia a los antimicrobianos transmitida a través de los alimentos.
One of the main goals is to support countries in generating.
Now, let me begin the session 4 of 5th MFDS GCFA. And this session is being held under the theme of sharing policy initiatives and outcomes by international organizations. And chair will be Professor Choi Jong-chan, and he is from Jeonbuk University, and he is a professor in Division of Biotechnology. And he is an expert in many microbiome expert, and he also his research interests include antibiotic resistance approaching with epidemiological studies in genome level, and now he served as advisory member of several government councils related with antibiotic resistance. So I'd like to hand over my microphone over to our chair, Professor Choi Jong-chan. Please welcome him with a big round of applause.
I'm Jong-chan Chair from Jeonbuk National University. It's my honor to take a chair in this session. We have a long journey for 2 days. Finally, this is the last session, okay? Don't take a lap. We'll proceed this session with 3 speakers officially. In third presentation, the 2 speakers will give us a talk. As like other sessions, we are going to have a Q&A time after 3 presentations. Let me introduce the first speaker, Alejandro Garcia, who is an animal health officer at FAO. He is a veterinary epidemiologist. He got a Doctor of Veterinary Medicine degree from the Complutense University of Madrid in Spain, and also got PhD from Utrecht University in Netherlands, related with AMR epidemiology. Since 2018, Dr. Garcia has served as an animal health officer at FAO. Please welcome him with a big round of applause.
Thank you very much, and thank you very much for giving us the opportunity to present the results on the INFAM system. So we presented the system for, I think already for 3 conferences. We've been presenting it from the conceptualization of the global information system through the piloting phase, and now we are in the early implementation phase. So today what we would like to show is the initial results of the implementation phase. So let me go back. So, and we would like to focus specifically on what the INFAM system is already teaching us. So we started the first open call for data in 2024. We invited all countries in the globe to participate, and we are going to present you today the initial results of this global open call. So just to give you a little bit of history, the current situation in many countries we have is this one. So we have a lot of data files all over the world, dispersed through different agencies, different institutions, universities, ministries. This actually can create a lot of reactions in humans. We can go from having despair, frustration, but all this actually presents a great opportunity. If we put all this data together in a harmonized way, and we consolidate it in a way in which we can transform it in information, we can make use of this data. And this is the conceptualization of INFARM, and that's why we started to develop the INFARM system to support the integrated surveillance efforts from the other organizations, from WHO, also from WOHA, but also to make a space and to get to a place with countries in which we can transform all the AMR data in food and agriculture into information that can be used for policy making, for evaluating the effect of interventions or policies on antimicrobial resistance, but also for stewardship, for antimicrobial use, good practice of antimicrobials. So just to give you an overview of what is the approach we're using for the surveillance system, we have a global information system which is composed of a digital platform, a web-based application for collecting of data on a harmonized and structured way, but also the activities that are supporting countries in collecting, collating, analyzing, visualizing, and effectively utilizing the AMR data monitoring and surveillance data. Primarily, we started with livestock, fisheries, aquaculture, and also food products. And currently, this year, we are also expanding the scope for the collection of antimicrobial use in the plant sector. So, as was said, it's not only the web application, but it's also the set of tools, access to networks, access to technical resources that are cascading down the implementation of international standards, WOHA standards, Codex standards. They offer a framework for implementation, but we need to provide practical solutions for them on how to implement these standards. And this is what we do with the INFARM system and with the specific tools. One of the flagship tools that we have in the organization is the assessment tool for laboratories and AMR surveillance systems, the ATLAS tool. ATLAS has been implemented in more than 50 countries so far, more than 200 laboratories in the world, and it's also providing a structured approach for measuring, sorry, it's moving along by itself, but it's providing also a structured approach for measuring the capacity of countries in a progressive improvement pathway. So we measure the capacities from 1 to 5 on what is the level of reliability also of the data we are going to receive in the INFARM system. So through the Atlas digital application that we are currently developing, and we are gathering to also contextualize the data that we are receiving in the INFARM system, we will be able to give a mark of quality or a mark of reliability of the AMR data we are receiving in the INFARM platform. So, and also just to give a little bit of context on what is the surveillance approach, in INFARM we propose a framework that is supporting countries in the harmonized collection and collation of data across different purposes. The purpose of animal health, for stewardship, for informing clinical decisions, but also the purpose of public health. So this framework is divided across these two main purposes. In the purpose of animal health, we are focusing on animal pathogens, bacterial pathogens, that are using veterinary diagnostic laboratories as the main surveillance site. So we are obtaining the isolates, and countries can look at the antimicrobial susceptibility testing and the antimicrobial resistant profiles in these animal pathogens to inform clinical decisions. Then on the public health side, we have all those programs that are identifying and monitoring foodborne antimicrobial resistance bacteria, and also indicator bacteria, E. coli and Enterococcus. And here, what we are trying to see in the public health programs is to look at what is happening across the whole food chain. So you can see that in these two purposes, animal health and public health, we have 5 surveillance programs. In the animal health, we have the diseased terrestrial animals, the diseased aquatic animals, and in the public health, we have 3 programs: healthy terrestrial animals, healthy aquatic animals, and food at processing and point of sale. So the INFARM system provides a framework for harmonized collection across these 5 different surveillance programs, and it offers a way in which we can gather the information and analyze it in a proper way so that we can extract epidemiological inferences. So just to give you a little bit of the journey that we were following on InFarm, we are currently in the early implementation and expansion phase. So in 2023, we did a pilot with countries of the IT platform. In 2024, we launched the digital IT platform, the first open call for data. And currently, we are finishing the early implementation phase and entering into the expansion. We are expanding to antimicrobial use data collection in plant production and protection. And we are also expanding to other areas on data management systems for facilitating countries the submission of data into the platform. In the data call in 2024, we got over 50 countries enrolled, 49 submitting questionnaire on implementation, and 28 submitting AMR data that comprise more than 500,000 bacterial isolates. So this is just to give you an idea of the volume of participation in the data call in 2024. We also did regional trainings on FinFam focal points, regional trainings in the Asia region, also Africa, and also Latin America, covering more than 200 participants and gathering more than 50 countries in the 3 regions. Thanks to these regional trainings, we were able to engage countries in the participation and to get these initial numbers of participation in the first open call, which are numbers that are very similar to the first open call for data that was happening for the global antimicrobial resistance surveillance system, the GLASS WHO system in 2016, I believe. It was when it was launched, and it got very similar numbers in terms of participation before the expansion. Right now, we are in the second annual open call. So in July, we opened the second open call for data that will run until December. So during all these months, countries can gather the data, can compile the AMR data files for submission into the system according to the requirements and technical specifications for the InFarm system. And then we will expand the system and we will consolidate. These are the next phases that you can see that are running up to 2030. So also this is also very important to understand that when we are establishing a global information system, we need time. A global information system is not set up in 1-2 years, requires a lot of time and maturation process so that it can start generating reliable and useful information for the countries, but also at the global level. So what I would like to go through the presentation and very quickly is on the report on early implementation. Currently, we are preparing this report, which is compiling data, the descriptives of the questionnaires on surveillance implementation, but also the descriptives on the AMR data that we received from countries. This, of course, is to be consulted with countries, and before we publish all the interactive data visualizations in the preview of the public interface. And we expect to publish this report by the end of this year or early 2026. So everything will depend on how long we take during the consultation process with countries. So important to mention here is that countries can participate in the InFarm system just by submitting a surveillance questionnaire. A questionnaire with descriptive information describing how they implement surveillance activities in their countries. So this is the initial step for participation and the only requirement. The next requirement for participation is voluntary, and they can submit also AMR data. But I want to make this distinction because the report is going to have these 2 elements, the descriptive analysis of the surveillance questionnaires and the descriptive analysis of the antimicrobial susceptibility testing data or AMR data. So just to give you a bit of an overview, right now this is the map of countries enrolled in InFarm in 2024. Across these 49 countries that you can see in the map, 28 of them submitted also AMR data. And when countries submit data to the InFarm system, they can submit it at 3 different levels of confidentiality. The level 1 is the level in which countries submit AMR data, but they don't want to make public their data to the world. So they just want to obtain the interactive data visualizations for their private use. The level 2, countries participate in the global reporting, but they don't want to show the identity of their country in their global reporting. This means that we cannot show the identity of those countries participating through the submission of AMR data. And level 3 are countries that are participating, like in GLASS right now, which are countries that are showing their identity in the sharing of AMR data. So that's why in this map, we only show countries that at least submitted the surveillance questionnaires. But we are not showing the countries that actually submitted the AMR data, because otherwise we would reveal their identity. So in terms of questionnaires, so we have 96 different submissions of questionnaires. We have questionnaires specific for each of the surveillance programs, and also a questionnaire that tries to capture information on how these programs are integrated. So most of the questionnaires were coming from Africa, and then Asia, America, and Europe, as you can see here in the numbers. And also, most of the questionnaires were coming from healthy terrestrial animals. This is not a surprise. We know that most of the surveillance that is being carried out right now in the world is on healthy terrestrial animals, and also is part of the foodborne antimicrobial resistance monitoring that is promoted by the international organizations. Then we have also a little bit of questionnaires on healthy aquatic animals, food and processing, but this is lagging behind on the most of the reporting of the questionnaires, but also on the AMR data, which is coming from the healthy terrestrial animals. And we did the analysis of these surveillance questionnaires. I'm not going to run into many of these details because otherwise I will not have time to present. But we did a stratified analysis of the questionnaires. We did a whole global analysis of all the questionnaires, regardless of the surveillance program. But we also did an analysis by surveillance program, or for the public health or the animal health purpose. And we also did an analysis across the different regions, by region. Today, I'm just going to give some highlights of the high-level results that we are preliminarily identifying in the analysis of the whole 96 questionnaires that we received, regardless the surveillance program. And we have 4 different domains. I'm going to go through these 4 different domains of the questionnaire, which are the surveillance network components, epidemiological design, laboratory design, and reporting of results. So, on the surveillance network components, when we talk about a surveillance network, we talk about 3 main components. The peripheral laboratories, responsible for producing a big enough amount of isolates for the surveillance system. Then we have the national reference laboratory that provides support to this peripheral network of laboratories on standardization, but also provides support on the quality assurance. And then we have the National Coordinating Center. The National Coordinating Center is something that in the results we are realizing that is not existing in many countries. These are some of the figures, some of the percentages we received. And the most interesting one, I believe, is the last one. We see that 49% of the countries have all 3 components established, meaning that more than half of the countries are not having these 3 components integrated. So basically we can say that they are not having a surveillance network which is structured. This is calling for the need to support the establishment and integration of surveillance networks with both combined microbiology and epidemiology capacities. That's why I'm putting this cable here. Basically, we have the data generation in the laboratories being disconnected from the epidemiological analysis and dissemination of information. This is the connection we need to start establishing in countries, and especially in low middle income countries. In terms of the epidemiological design, we confirm through the analysis of the results that countries are using active surveillance for healthy terrestrial animals, not surprising, but it's a good confirmation, and passive surveillance for terrestrial and aquatic animals. We also realize that there is a moderate level of statistical representativeness of the data. When countries submit data to the INFAM system, they also need to tell us what is the level of statistical representativeness. We see that half of the countries report having national surveillance program, meaning that is representative of the national situation, but many countries actually just have pilot activities, point prevalence surveys, or small pilot scale studies. And then also countries are using internationally recommended target microorganisms, not surprising again because there is a lot of international guidance. There is also the Codex guidelines for integrated monitoring and surveillance of foodborne AMR. And this is not surprising, but it's a good confirmation. Good confirmation that most of the countries are focusing on E. coli, Salmonella, Campylobacter, Enterococcus. And then also many countries were defining panels, specific panels, and this is what we show here. I don't have time to present today all these, but these are the defined panels that countries were using in terms of following the WHO highest priority critical important antibiotics. Sorry. So, in terms of laboratory methods, we also saw that there is a high level in the use of conventional methodologies. This is also something very, very interesting, because the other day when we were in the table having dinner with my colleagues, we were talking about whole genome sequencing, and we were also trying to understand how these technologies can be transferred to low middle income context. The reality is that 80% of the countries that are participating in the system are using this diffusion. So actually they are using the most basic microbiology technique to produce the data. This is calling, and also is making evidence of the big technological gap that we need to face in the capacity building for antimicrobial resistance monitoring and surveillance. On the other side, we also have high levels in the use of international standards. 77% of the countries use CLSI standards, and then there was a smaller percentage using EUCAST. I'm not going into the table, this is in the manual for implementation, but these are the recommendations on interpretation criteria to be used according to the different surveillance programs. We also confirm that countries are using these recommendations that we provide in the manual for implementation of INFARM for interpreting their results. And the reporting of results. Many countries said that they have a responsible unit for doing analysis and reporting. But, and I put it here at the end, only 57% of the countries reported producing reports on a periodic basis. So this is like a broken speaker. You have the speaker, but nothing is coming out from the speaker. So we need to support the analysis, interpretation of results, and the national reporting. And this is also what we are trying to do through the system with the interactive data visualizations. Then I'm going to move into the next part of the results, and I realize I have a very short time to present, but I'm going to be very quick. In the visualization of AMR data, I just want a word of caution here. This is subject to change, we are still analyzing it, and I'm going to present some figures that are just for examples. And I'm not going to say if this is coming from E. coli, from broilers, from poultry. So this is all data just to show you what is the potential for reporting and what you can expect in this first global baseline report, but also to see what we can expect in the future. And also these visualizations are just produced with level 2 and level 3 data of confidentiality. So I explained before, we are only using countries that agreed on sharing at the public level, showing their country identity or not showing their country identity, but countries that agree in sharing this data to the public. Most of the data coming from E. coli in healthy terrestrial animals, from broilers and from pigs, which is the main commodities across the world. And if we look at this data, this is a picture of the web application. In the web application, countries have access to a private interface. And they can get access to different interactive data visualizations of their data. We provide general information on the profile of information they are sharing into the system, the number of surveillance sites, number of isolates. And then we also provide an AMR analysis and trend dashboard with tables and also graphs on absolute and relative frequencies of antimicrobial resistance. This is the normal classical descriptive analysis of antimicrobial resistance, but countries can get access to this information in the web application. What we also have in the application is what we are analyzing now, is we have multi-country visualizations. Visualizations in which we pull all countries together, and we have 3 multi-country visualizations, the participating countries, the map that I showed you before. We have a map with prevalence of antimicrobial resistance. This map, I believe, will not be possible to produce until we have 2 or 3 cycles of open calls, until 2025 or 2026. And then we have the median of AMR proportions across countries. So I'm going to show you just median of AMR proportions just as an example. Here you can see an example of a box plot. For those in the back, you cannot see, here are the antibiotics. And here is the prevalence of the median of AMR prevalence or AMR proportions across multiple countries. So each point is a country, which is, of course, anonymous. And then we have the distribution of the prevalence of those in these countries. And then, of course, we have also the median, which is in between this orange box. You can see this, of course, this is raw data in healthy terrestrial animals, but this is not possible to extract any conclusion from here because this is mixing poultry, pigs. This is actually, epidemiologically speaking, does not make sense. So we need to start filtering. That's why in the system we have a lot of filtering options for a specimen, origin, microorganism, the biotic class. When you start filtering, you realize that the box plots become much smaller, of course, but also the number of countries from which we can extract epidemiological inferences is much lower. So we start filtering by animal species, specimen, other metadata. So this is like a Russian doll. So it looks like we have a lot of data, but when it comes to the actual interpretation of the data, we still need to get to more cycles of data production to start having an adequate volume in this categorization. So in poultry, E. coli, feces. So if we use all this combination of metadata, we still need to produce more cycles of information. This is just to give you an idea, but of course this analysis of box plots can also be stratified by high-income, low-income countries, by regions. It can also be used for benchmarking what is the national situation against the regional situation or the global situation. And this is where it comes the benefits for countries. Countries can get access to a safe space to store AMR data. They can also submit data across the whole food chain from multiple institutions, from food safety authorities, from Ministry of Agriculture, and also they can submit across different purposes, animal health and public health, as I was saying before. They can also get access to the interactive data visualizations, which in low middle income countries is a big challenge with a big lack of epidemiological capacity. Then also they can get access to a centralized repository where to upload the data, the results of the assessments through the Atlas tool and surveillance programs. And something that is very important in the system, that is improves the coordination between authorities. In the system, in the private interface, the focal points that are designated for the different surveillance programs have access to a national space, meaning that if the food safety authority is submitting data, the focal points of the food safety authority will also be able to see the data that is submitted by the Ministry of Health doing surveillance in healthy terrestrial animals and in primary production. So this means that it connects the different authorities, the focal points across the different authorities in one single space to see what's happening upstream and downstream the food chain, and also to comply with the implementation of Codex standards. And just to finalize, what are the next steps? We will finalize and publish the first report of INFARM by the end of this year. We will expand the coverage to new surveillance programs. We need to put more emphasis on collecting data on fisheries and aquaculture. We need to put more emphasis also on the animal health programs for bacterial pathogens to inform stewardship. So there is a lot of emphasis to put across different areas beyond the healthy terrestrial animals without losing focus on that as well, of course. Also support the expansion to new technologies, to whole genome sequencing, to molecular epidemiology. So how we can incorporate molecular information also in the global reporting. We will support countries in integrating the surveillance system into policy and action. That's why we also support countries through the INFARM action. We also support countries with the establishment of national surveillance plans and strategies. These national surveillance plans and strategies are key to ask for budget. Right now, there is a consistent lack of budget at the national level in national action plans to do surveillance. This is expensive. So the first thing is to have the strategy, the national surveillance strategy, endorse it by the government, and move it to the next level in asking for budget. We also support on that. And of course, rely on the established communities. This is for sustainability and beyond the life of projects. We request countries to rely on the INFAM communities, on the Atlas community of assessors, and also in the establishment of national coordinating centers for epidemiology. So thank you very much. I would like to close just with a word of thank you to the government of Korea and to the Ministry of Food and Drug Safety for funding this project for all these years, and also to make possible to establish the foundations of this global information system. And thank you very much.
Thank you for a nice presentation describing INFAM system. Let's move on to the next presentation. Next speaker is Dr. Muhammad Usman Jahir, who is a regional AMR surveillance and One Health specialist. He got Doctor of the Veterinary Medicine degree from University of Agriculture in Faisalabad, Pakistan. I've been there 10 years ago. And also got PhD from Colorado State University in USA. He has many experience for AMR surveillance, especially he involved in establishment of guidelines related with the national surveillance strategy for antimicrobial resistance in Pakistan. Please welcome him with a big applause. Please.
Thank you so much and good afternoon. And first of all, I would like to thank the Ministry of Food and Drug Safety in the Republic of Korea for the invitation for me to come here. And I'm presenting here on behalf of FAO's Regional Office for Asia and the Pacific. So I'll not try to take too much of time. And my presentation is not too much around the research work. Rather, it's about how we are supporting the countries in the Asia-Pacific region for AMR surveillance in agri-food sectors. And I will give some highlights from sector-specific and integrated surveillance of AMR. So before I start, I'll try to play with this. Okay, it worked well. So, yeah, I'm not good with it. So we know that AMR is an issue that does not recognize any physical, biological, or sectoral boundaries. When it does not recognize these boundaries, so it means its drivers and transmission pathways are in different sectors and different disciplines. And these disciplines are often segregated. And they are generally governed in the countries in very different ways. So every sector, every department has their own mandate, like agriculture, environment, livestock, food. They have their own mandate. The same goes for the human health. So everyone has their own mandates. But addressing this issue, the bugs that don't recognize any barriers, requires a One Health approach. That we already know. But once we say One Health, that also means that we need to strengthen each sector as well. We cannot achieve what we want to achieve in terms of combating antimicrobial resistance unless we strengthen respective sectors, because each sector has their own mandate. They have their own expertise. So expertise of specific sectors in their specific mandates will actually help us achieve the containment of antimicrobial resistance, which is a silent pandemic as well. So we need to, this also leads to the sectoral accountability as well, that each sector has a role to play, each contributes to achieving containment of AMR. I'll not go through this image. I think you have seen this image and images like that in the past, but just to give you an idea how it's all interlinked. It's like a wicked problem, but we got to come up with innovative ways to address it. So just taking you back about a year and a week. So last year in September, UN General Assembly high-level meeting on antimicrobial resistance came up with over 100 commitments. There were several around antimicrobial resistance surveillance. I'm only giving you 2 here. And again, I will not be reading everything. I know you can better read it. You can also refer to it later on. But the idea is that one of the commitments says that strengthen the national capacities for AMR surveillance and AMU surveillance data. that coming from sustainable, sector-specific, integrated, and interoperable surveillance systems for different sectors in humans, animal, environment. And also it talks about encouraging the countries to report the quality data on antimicrobial resistance and use by 2030 to the existing global AMR surveillance systems such as WHO's GLASS, WOHA's NMUs, FAO's INFARM, so that these data can be ultimately used into the GISA. So about GISA. GISA is under development. It's a global integrated surveillance architecture for antimicrobial resistance and antimicrobial use. And it's planned that it will take data from different databases, the different global surveillance systems such as WHO GLASS, which is for the AMR surveillance and AMU monitoring in the human health sector, WOHA's NMUs, which is for AMU monitoring in the animal health sector, FAO's INFARM, which is for AMR data from food and agriculture sectors, as well as antimicrobial use from the plant sector. And last but not the least is the environment sector for AMR as well as the residues. And this all feeds into the GISA, and the quadripartite organizations are working on it. I don't need to go into too much details. Jorge gave a very fruitful presentation yesterday that we had a lot that we learned from it yesterday. Now going into a little bit of what FAO is doing. FAO's work is anchored around FAO's action plan on AMR. And our vision is that we want to ensure that food and agriculture sector dependent economies and livelihoods are made resilient to the impacts of AMR. That's what we want to do. That's how we are supporting our members in the region as well as globally. So how we do it, we have action plan has 5 pillars. Those are around stakeholder engagement, surveillance and research, good practices, responsible use, and governance. And through this, we want to ensure that AMR prevalence is reduced and its emergence and spread is also slowed down in the agri-food sectors. As well as we want to ensure that antimicrobial efficacy is preserved while ensuring that sustainable food and agriculture production. We also want to balance that as well. Going into the surveillance component, FAO is supporting its members through the tools that FAO has for strengthening the national capacities for AMR surveillance. FAO also supports the members through the field interventions, training programs, as well as the laboratory networks that we have, that we are supporting our members for the establishment of AMR lab networks at the country level, so that there is a cross-learning between different sectors. I'll give you more details about that in the coming slides. In the Asia-Pacific region, we have several ongoing projects funded by different donors. We have project funded by the Fleming Fund, which is the UK AIDS Department of Health and Social Care project. We have a project funded by the Ministry of Food and Drug Safety, Republic of Korea, which is on the Codex standards and implementation of Codex standards to contain and reduce foodborne AMR. And then we have a project funded by the European Union, which is also working across the region. More details will come in the next slide. But once we support our members in the region, we have a regional strategy as well. There are 5 main elements for that. We start off with understanding the context. What is the situation in the country? Then we apply the tools or develop the tools. We deliver the capacity building trainings to the countries. We also foster the collaborations and partnerships. And then we ensure that countries' systems are sustainable so that there is long-term implementation of the activities so that we can have the meaningful impact. Again, a little bit more details around the surveillance component, how those 5 elements that I presented of the regional strategy in the last slide, how we are doing it. In terms of understanding the context, FAO has its assessment tool for AMR surveillance and laboratories in agri-food sectors called Atlas. Many of you may have heard it. So that is, we deploy and roll out that tool in the countries to assess where the country is. And the tool helps us identify the key interventions country may need to do to have a better system for AMR surveillance and lab networking in the country. I'll not read the other stuff that's on the slide for the bullet number 1, but for the second one, we also support the members. Okay, now we know the context. Okay, country need to develop a surveillance. How do we support the country or our member? We develop series of guidelines. More details will come later. And we also develop the guidelines and help the country set up a surveillance system through a consultative process as it fits the country needs. Then also supporting the countries with the capacity building trainings. Those can be on the surveillance design, trainings around the laboratory methods, diagnostics, as well as AMR data management. Those trainings FAO delivers and builds the capacity of our members. And then also FAO has a network of reference centers that FAO leverages their expertise. For instance, in the region, we have 2 reference centers. One is in Thailand, Chulalongkorn University, and the second one is in China, China Agriculture University. These reference centers provide support to members as they in their areas of work, areas of collaboration, and all the information is available on FAO website. And currently we have 10 reference centers. Of course, 2 are in the region, rest are spread across the world. And then we also ensure sustained implementation. How? FAO also helps the countries to, once the assessments are done, for instance, as I mentioned in the first bullet, we don't stop there. We help sit with the country, identify the key areas and interventions and the priority sectors for the country where they want to do that work based on their resources so that the activities are sustained. Also help the countries to update their AMR national action plans so that food and agriculture sectors are represented. You may be aware that Global Action Plan for AMR is currently being updated, and it's important that food and agriculture sectors are at the heart of that process so that their needs and priorities are identified so that we can address this issue of AMR equitably. These are some images to make it more fancy and engage people. On the right side of the screen, no, it's your left side. So on the left side of the screen, you see a series of guidelines that we are developing in the region to support our members for AMR surveillance. We also conducted a workshop in Bangkok last year. More details will come in a few slides later for the benchmarking the AMR surveillance across sectors in humans, animals, and the environment. And we had over 100 representatives that came together to discuss what is the status of AMR in different sectors. Also supporting our members with informed trainings. We conducted a training this year in July in Bangkok so that FAO members from the region have the capacity to manage their data, to handle their data, and prepare their data and report it to Inform if they wish to do so based on different options that are available, as Alejandro mentioned before me. And also supporting our members in the training programs for the labs, trainings around the whole genome sequencing, training around the avian pathogenic E. coli, and so on and so forth. And also the proficiency testing schemes provided to our members so that the data that is generated, its quality is also ensured. So going a little bit further, not spending too much time on this one, but once we dissect it, we take it out, we see the animal health sector has connections with the human health and the environment. Of course, animal health also includes aquaculture. Oh, 1 minute. Seems like the update did not show up correctly. Okay, 1 minute. Okay, so I just wanted to bring everything here. So once we talk about the animal health sector or the livestock sector, for instance, we have to think about pathogens that are not zoonotic, but are important for the veterinary sector or the animal health, so that the veterinarian has the information on the antimicrobials that they need to prescribe so that the animal health and welfare is not compromised, as well as animal production is not compromised. That's one angle that we work on in FAO. And then also, in the public health concept, connection, animals are produced for food, and those food animals may have the pathogens or the bacteria that may be harmful to humans. So we also work on that aspect as well, so that the food that is produced from the agri-food sectors, it's safe. And also around the environmental connections, farms produce, say, manure and slurry. That may be going into the wider environment. The same may be used for irrigation purposes. So we also work on that interface as well, so that we are developing a guideline that I'll talk later about AMR in the food animal production environment, aligning it with the Codex standards. And also the aquatic pathogens and the antimicrobial residues. So, as I mentioned in the previous slide, so there are these 6 key areas in terms of the developing the guidelines that we are working on and supporting our members around AMR in the healthy animals, which is for the public health purpose, antimicrobial residues from the food of animal origin, pathogens of veterinary significance, which is for the livestock purpose or the animal health purpose, AMR in the aquatic animals, AMU monitoring and quantification at the farm level, and last but not the least, AMR in the food animal production environment. So these are the 6 guidelines, as I mentioned. The guideline 1 was published in 2019. Guideline 2 on AMR in the bacterial pathogens of veterinary significance or the diseased livestock is being finalized and will be published soon. The guideline 3 on AMR in aquatic animals was published last year together with Singapore Food Agency and National Parks Board of Singapore. Guideline 4 on AMR in food animal production environment is being finalized. We are hoping to publish it in the coming year. And then AMU monitoring at the farm level, guideline published together with VOHA and FAO. And the last one is antimicrobial residues in foods of animal origin, which is also being in the final stages. Some of you, I think, may have participated in the consultation that was done in January of this year. So a little bit going further into the AMR in the healthy animals, we did not just develop a guideline and that's it. We went and supported our members in terms of how they can now design their surveillance, identify their priority production systems, priority animal species, and then identifying the national capacities, which pathogens, bacterial pathogens they can start from, say E. coli, Campylobacter, Salmonella, or Enterococcus, and then also supporting the countries in terms of the lab trainings, epidemiology trainings, data trainings, so that they have the national capacity that they can utilize for containment of AMR. And then in terms of the AMR in aquatic animals, the guideline was developed together with National Parks Board in Singapore and the Singapore Food Agency last year, finalized and published. And this guideline is also helping the members in the region and across the world to have established AMR systems, surveillance systems in the aquatic animals. And a training was also conducted this year in Singapore to support our members on the lab methods. Where does all of this work fits? FAO launched, we are talking about AMR, but we have to reduce the need for antimicrobials on the farms. We heard about antimicrobial alternatives. We also heard about other presentations yesterday. So FAO has launched the initiative for 10 years called RenoFarm, which is reduce the need for antimicrobials on farms for sustainable agri-food systems transformation. It has 3 targets. The first target is that 100 countries participate in the initiative with their AMR national action plans fully implemented in food and agriculture sectors. Through our work for the assessment of AMR national action plans in the food and agriculture sectors and their implementation, we have identified that national action plans are often do not necessarily always equitably represent the food and agriculture sectors. So that's very important priority for the organization, that national action plans have the food and agriculture sector components and those so that those can be implemented. And the second one is about 50% of the workforce is trained on good practices so that the veterinary and agricultural workforce has the training on good production practices. And the last but not the least is 80% of the participating countries report their AMR data to InFarm as well. And RenoFarm was highlighted as one of the key initiatives to reduce the need for antimicrobials on farms, together with VOHA's list of veterinary important antimicrobials in the UNGA high-level political declaration last year. InFarm, I think I will not spend too much time on this slide, but Alejandro gave a very good presentation on this. Countries participated. The next call is open. FAO is working towards generating a report on that as well. Okay, some highlights of the key activities in the region that are supporting the countries in terms of integrated surveillance and integrating across the sectors and better understanding of the situation. We conducted a benchmarking workshop last year. Korea participated along with representatives from other members in the region, quadripartite from the global, regional, and the country levels, reference centers from FAO, WOHA collaboration centers and WHO collaboration centers, and other international experts. They came together, discussed what is AMR surveillance looking like in countries. More often than not, when the meetings happen, those are generally for one sector. But this meeting brought together people from 3 sectors in the country. Within the animal health, it's also brought together people from terrestrial and aquatic both, so that we can hear from the countries what are their priorities. And in terms of integration, one of the big thing that was highlighted that we know already, but it was again re-emphasized that it's a coordination that is the main problem in terms of integration. And of course, there are issues around the capacity of the country. There is no priority, for instance. Who leads, it's also a problem. So the workshop came up with a lot of recommendations about integrated surveillance, that we need to go to the country level. Integration may not be one size fitting everyone. It's every country's needs are different, their priorities are different. So now we are working with the countries. We conducted a workshop in Malaysia a few months ago to have a national level landscaping and situation analysis of integration, how and where the integration for the country can happen. Does the country have the resources and the capacity for that? If not, what is exactly needed? So that we did that. We are also going to do that, a similar exercise in Korea in the afternoon today, and then also in India in the coming months. And then some of another activity that we are supporting the countries, as it came out from the workshop recommendations, is supporting the lab networks in the countries, so that different sectors can learn from each other's sectoral expertise. And I think yesterday there was a discussion about the data use. This also came in the benchmarking workshop last year. Countries have a ton of data, but they sometimes don't know what is the utility of their data. So we are going to establish a regional technical working group, which is expected to be launched towards the end of this year, which will bring together experts from different countries with expertise in different areas. The main purpose is it will provide a platform for the countries in the region and for the countries as well as the experts from different countries at the quadripartite and other organizations, so that they can discuss how integration can happen, how they can use their data. So I'm not going into the details of membership and others, but we are planning to anchor it with the regional quadripartite in the Asia-Pacific region. Okay, so briefly about the project funded by the European Union. You may have noticed that I showed you 3 projects in a slide maybe 10 minutes ago. But here, I'm quickly walking you through the project funded by the European Union. It is working with 9 countries. 2 countries are the partners. Republic of Korea and Japan are the partner countries, while 7 other countries are the beneficiary countries. We are building country capacities for multi-sectoral AMR national action plans, AMR and AMU integrated surveillance, private sector engagement and communication, research and innovation, and pandemic preparedness through a One Health approach. And the work that I presented earlier about the benchmarking workshop, that was conducted by the project funded by the European Union. We also have a project funded by the Fleming Fund, which is the UK aid project. This project started in 2021 and expected to end in March of next year. Again, this project is taking a One Health approach to support members in the region. It is working in 5 countries in the region, but it's also working in sub-Saharan Africa through our regional office in Africa. So it's also supporting the countries in generating the quality data so that that data can be used, and also developing economic cases for investment in AMR as well. And this project is also supporting countries through Atlas tool, FAO's progressive management pathway tool, to support the development of national action plans that are representing food and agriculture sectors. And then we also have a project funded by the Republic of Korea, Ministry of Food and Drug Safety, MFDS, which is also the host and organizer for this conference. So this project is also working in 4 countries in the region, in the Asia Pacific region, while 2 countries in Latin America in the Caribbean region of FAO. This project is mainly around implementation of the Codex standards for containing and reducing foodborne antimicrobial resistance. This project is mainly working around 3 pillars: awareness and advocacy, governance, and AMR surveillance. So this project has helped countries in reaching in elevating the profile of AMR through awareness raising and advocacy with veterinarians, farmers, policymakers, and youth, and also help the countries in strengthening their AMR surveillance system through the assessments, through the pilot studies on foodborne AMR in countries like Nepal, Pakistan, Mongolia, and Cambodia, and also help the countries in terms of strengthening the governance system for AMR National Action Plan, updating and development such as in Pakistan, as well as legal assessments and other support that this project is providing to the countries in the region. So with that, we are not doing it alone. We do it with so many partners and collaborators. So we are thankful to them as well for the support and our collaboration. And also we thank the different donors that are providing the money to FAO to support its members, such as EU, Fleming Fund, as well as Republic of Korea, and more recently the Pandemic Fund as well. With that, thank you so much.
Thank you for a nice presentation about the surveillance system. So we are out of the time. So let's move on to the next presentation. As I told you, 2 speakers will give presentations, right? Next speaker is Mr. Mohan Krishna Maharajan, who is a Senior Food Research Officer from Department of Food Technology and Quality Control under Ministry of Agriculture and Livestock Development in Nepal. Now, he is in charge of sanitary and phytosanitary national inquiry point in the department, as well as spokesperson. Please.
Brief introduction of me. And first of all, I would like to sincere thanks to the Ministry of Food and Drug Safety, Republic of Korea, as well as all the team of the ACT project for supporting us in working with foodborne AMR, as well as the opportunity to present here this national experience of Nepal. So in my presentation, I will just cover briefly on the background and food safety related to the foodborne AMR and some brief introduction to the Food Safety Authority and food sector of Nepal and contribution to the food sector addressing foodborne AMR, as well as some AMR activities and programs and its partnership and collaborations. If we see in brief background of Nepal, the total population of Nepal is 30 million, and 2/3 of the population is engaged in agriculture, and the livestock population is booming up, and so the issues of AMR is growing up as in context of global scenario. As we talk about the issue of AMR is rising in global, same as in case of Nepal also, it's the top 3, the issues of burden on foodborne AMR. If we see the data of recent data of foodborne AMR, that is around 5,000 death cases attributed to AMR, and there is so vulnerable to the young children, and there is massively misuse of antimicrobials in all sectors, and the integrated monitoring and surveillance system for the foodborne AMR need to be established firmly, and we do have NAP AMR in action. However, we need the dedicated budget for the effective implementation, and still we do have the AMR regulatory gaps. If we see the development of NAP AMR, that we have started it since 2011. Now we do have NAP AMR endorsed in 2023. 5 years NAP AMR, which is from 2024 to 2028. And the development of National Action Plan of AMR is aligned with the global, so that we do have also the 5 pillars. I would not like to elaborate it. If we see the governance structure of the AMR, governing structure, we have high-level multi-sectoral steering committee, and we also have National Technical Working Committee, and also the technical working committees in different strategic priorities. And we have sectoral technical working groups, especially in human sector, animal sector, food and feed sector, and environment sector, and they are doing their respective field in their respective activities. We have, in case of Nepal, we have initiated some key intervention in foodborne AMR. So in case of AMR, in the public health sector, there is some kind of decision of red line for the antibiotics. And we also follow the recommendation of WHO. And recently, as a Food and Feed Safety Authority of Nepal, DFTQC, we have recently prohibited for using of antibiotics in poultry feeds. And we have a decision of using cholesterol in animal use. There is the support from quadripartite counterparts for international counterparts. We are getting the support from these counterparts, and we have national government counterparts in different division, in different ministry, in 4 sectors. And we have National Action Plan, which is also feasible for the provincial level, but still the NAP AMR can be conceptualized in the province at a provincial action plan AMR. If we are talking with the AMR surveillance in human health sector, they are quite leading sector in Nepal. So 26 Sentinel sites is collecting the AMR data, and that is sharing with the glass platform. And the human health sector is working together with One Health approach in the joint surveillance system recently. Same as human sector, the animal sector is also following. The Central Veterinary Laboratory, CVLs, working together with the regional laboratory, is collecting data, and they are also taking the surveillance, active surveillance and passive surveillance in the human as well as animal sector. So animal sector is also leading with the human sector. When we talk about the food safety sector, the Department of Food Technology and Quality Control is the responsible agency for food and feed sector quality and safety. So the department is also the Codex contact point as well as InfoSan emergency contact point. So we have initiated under the support of ACT project working on foodborne AMR. If we see the National Food Safety and Quality Management, the department is working based on the 4 pillars, that is legislations, inspections, and laboratory surveillance, as well as information, education, and communications. If we see the framework of policy and regulatory framework, these are the major regulatory framework for food safety and quality regulations. We do have National Reference Laboratory under the FTQC, which is accredited by ISO 17025. Now the department is working the extension of accreditation scope in microbial residue. If we see the journey of foodborne AMR, we have initiated the passive surveillance of AMR under the support of Fleming Fund project. So after kick-off the ACT project, now we are moving quite ahead, and now we are making surveillance active as well as passive surveillance And we are also doing it joint surveillance and strengthening the laboratories. When we talk about the ACT project, so we are working on foodborne AMR, especially from the support of ACT project funded by the Ministry of Food and Drug Safety, Republic of Korea. So we are very thankful to them and thanks. And especially these activities, major activities are the awareness and recognition of the Codex, and advocacy and awareness is a major activity. And similarly, the AMR surveillance in poultry value chain, and there are AMR surveillance initiated in the aquaculture, and some capacity building of institutional, and also we are working on capacity to enhance the antimicrobial residue testing. And not only the ACT project, we do have also the Fleming Fund project, which is focused on renovation of laboratories. So these two projects are working, but they are not duplicating the programs, but they are synergizing each other. When we talk about the ACT project, we have quite a lot of awareness and advocacy programs under these ACT projects. So there is quite a lot of awareness program in government officials. Similarly, the academic institutions, a lot of veterinary, agriculture, and microbiology and food technologies students are make awareness on advocacy on the foodborne AMR and Codex. And we also have the awareness program in poultry and aquaculture stakeholder, and we have supports for the World Food Safety Day program as well as World Antimicrobial Awareness Week. There is obviously some activity is developing the awareness materials within local language. It's in Nepali language, so these awareness materials has developed under the support of ACT projects. Similarly, the importance activities, one of the important activities is the assessment of the laboratory as well as the National AMR Surveillance System based on the FAO Atlas assessment tools. Based on the gaps of this assessment, the project activity is supporting on hands-on training of ACT project as well as some supports in the proficiency testing for the AST. So this shows the competency of the laboratories. And another important activities of the ACT is in the first times in Nepal, the two sectors is working together. One is food sector authorities activities as well as the animal sector authorities. So the AMR surveillance in poultry value chain has initiated as a first phase which is recently completed, and we are doing the joint AMR surveillance activities throughout the chain of feed. So the feed industries and processing plants is surveillance is carried out by the food sector, and the hatcheries and commercial poultry farming and slaughterhouse and meat shop is taken care by animal health sector. The preliminary reports, there is the issue of the multi-drug resistance isolation is there in the preliminary report. We will definitely submit the final report as soon as it's completed, the final report. So this shows the issue of EMDR isolates as well as it also shows where is the vulnerability in the chain. And this is also shows the issues and vulnerability in the value chain of the animal health sector. And another important part is that by the ACT project that we are jointly working with the academic institution in the surveillance of AMR or surveillance in the aquaculture with the collaboration with academia. And we would like to thanks to the ACT project that we are first time in Nepal. We are collaborating with the academia and some piloting in pocket area of fisheries. We are conducting this surveillance of antimicrobial use. And we have not only targeted the ACT pathogens. We also see the resistivity AMR genes which is concerned with public health. And we have another activities conducted by ACT project is a pilot study of CAP analysis, and it shows that awareness, only the awareness is not very effective. Sometimes the farmers, this study shows that the farmers which has knowledge about the AMR and the farmers which do not have knowledge of AMR, they are not doing significantly difference. So we need some kind of policy interventions. So the policy interventions is prevail that the report showed that the AGP used by farmers is remarkable decrease by 2%. Initially it was 15%. So some kind of policy intervention, regulatory interventions needed to make the effective in behavioral change, this data shows. But still there are, in animal health sector, they are using colistin. The project activity we do have by the ACT project is we are now strengthening the laboratory for the antimicrobial residue testing. So, and this LCMS is a gold standard for residue monitoring. So we are preparing for the laboratory strengthening, as well as we are planning to get the hands-on training for testing LCMS. So after these activities, we have planned to piloting the residue surveillance in feed and food product. Based on that, we are planning to set the MRL for the veterinary drug residue, which is the mandate of the departments. There is the assessments on AMR regulatory framework based on ACT tool. It shows that the different gaps are found, that the box is highlighting that the gaps. So we have to work on these highlighted area in coming days. So we have the regulatory policy regulation for the AGP, using of AGP, and so we are now needed for the policy intervention in labeling and withdrawal period and recording and reporting, as well as wastage management. So we have to work, and we would like to request for the continuation of the project working in this area to combat with the AMR. These are the INGOs and NGO partner working in AMR in different sector, animal health sector and human health sector and foodborne AMR sector, and especially the ACT project and Fleming Pond project is supporting for foodborne AMR. Now we have also the project of Fleming Pond, which is also supporting for the renovation of regional laboratory, not only in the central. For the regional laboratory, we are expanding the laboratory capacity in the region too, for the surveillance of the foodborne AMR. So this Fleming Pond is also supporting us in the foodborne AMR, but the activity are not duplicating with ACT, because some activity is supporting from Fleming Pond and some activity is supporting from the ACT projects. So they are synergizing each others. And our focus, the department has focused to setting the MRL aligned with the Codex guidelines for the policy implementations. And also we have a NAP AMR, but still we need the dedicated budgets to work on effective implementation of the NAP AMR. So if this project have made the foundation of the AMR, then this NAP AMR will be moved smoothly. And we recently have Food Safety and Quality Act 2024. This is the Nepali calendar, 2081, but in English this is 2024. So based on that, we are now under the process of developing different regulations, incorporating the food safety as well as AMR regulations. And we are now working together with the private sector, so we cannot go ahead alone with only the public sector, the government sector. We need to bring together with the private sector, so the building up capacity is needed. The ACT project also now working to making awareness and the building up capacity of the private sector. So we would like to go together with public and private sector. And the ACT project's lesson learned is to make the behavioral change, to adopt the code of practice developed by the Codex. Now the GLASS, that AMR data of public health is quite long ago. It's interconnected with the GLASS, as well as the animal sector is also sharing data upon enemies, but from the in-farm, so the DFTQC is now working to share the data of AMR and AMU. For that recently the department has assigned a focal person. So we hope that by the end of this year, we will also share the data. Not only share the data, this sharing of this data is also useful for us to interpretation for the policy intervention in coming days. So we will get the support from the Alejandro sir for the sharing of data and information through InFarm in coming days. So finally, I would like to convey my sincere thanks on the behalf of department that the support and giving platform to sharing the national experience of the Nepal towards the foodborne AMR. That I would like to thank the Ministry of Food and Drug Safety, the Republic of Korea, and always as well as the Secretariat of 5th MFDS of Global Conference, and also the organizer and event host, and also to the whole team of the FAO ACT project, from the team of the headquarters, team of regional, and team of national team. Here we have the national project coordinator, Dr. Sakar, and the Myeon Singh Choi, and the whole team of ACT project. I would like to thanks on the behalf of the FTQC that we have initiated the foodborne AMR in the department under the support of this project. So once again, I would like to thank. So this is, we have no time, so act now, stop the foodborne AMR. With this, I will conclude my presentations. So I will share after in discussion and questions.
Thank you for a nice presentation for about the current status in Nepal. We don't have time, but right now, we'll have a Q&A time. It's quite a little bit, okay? Please, come up to the table on the stage for the speakers. We have already 3 online questions, okay? So I'm gonna take just one question from the floor.
Thank you very much. If you allow me, I have one comment to Nepal, Dr. Mohan, and one question to Alejandro. Comment to Nepal. Thank you very much for your sharing your experience about the ACT project. We are really happy to see ACT project could catalyze activities, and now government has the leadership. We are really happy to see the advancement. And I have one question to Alejandro. Thank you. First of all, congratulations for the successful implementation of InFarm. InFarm could give a global AMR view in animal and food sector. InFarm, the most important thing is submission data from countries. I know several countries who have data but still hesitant to submit data because I believe they are not sure about the data protection. When they submit the data, they can be fully protected. can you give me another comfort to submit the data? Thank you.
Thank you, Myo-Sing. And I think it's a very important question. In FAO, as any other UN agency, we follow the data protection and policy rules. So we are under very strict rules for data protection for our members. All countries that are a member of the organization, they are subject to these data protection rules when they submit the data. We also have the terms of use for the INFARM system and IT platform that focal points need to agree upon the submission of data. So within the web application, countries need also to accept these terms of use. But basically, in a nutshell, the countries own this data. The countries are the owners, and they have the right over this data for publishing and also for removal from the public space. But of course, we will publish reports, written reports, with those countries that accept to publish at the global level. Something that is also very important is for countries to be reassured that we will always respect the level of confidentiality they choose in the submission of data. So if they choose to submit AMR data files at level 1, we will respect this. We'll never make this data public. And if they choose level 2, we will use their data for global or regional aggregations, but we will not share their identity. And if they use level 3, we will use their identity and we will show it to the public space. And a final point, also to encourage in terms of data submission, is also the reassurance to countries that AMR data in food and agriculture should never be a barrier to trade, should not have negative implications. Also, this is something that is very well also established also in the Codex guidelines for integrated surveillance of foodborne AMR. Is one of the first preambles that they include there, meaning that countries should not be using AMR data as a barrier to trade, but of course we will always protect countries for any positive negative impacts, any possible negative impacts on their production systems. And a final remark is also that all the data we collect in INFARM has no personal data. So we cannot identify the farms, we cannot identify, it's a global information system, it's not a traceability system. So as a global information system, we will collect data at the national level, but it will be not possible to trace it back to the original of a specific farm or a specific person. Yeah.
Thank you for the question. Yeah, that's, we do have the national AMR now, so government is now implementing side by side. So this project is making the foundation and a lot of informations and evidence that will definitely works on for giving the priorities of the national annual program. So DFTQC is now incorporating the foodborne AMR with the national annual program with the government of Mazet. So we would like to request to help some years so that when there is a foundation, then definitely the government Nepal is willing do as per the national AMR, NAP AMR.
Yeah, we're going to take one additional question. Yeah. Please.
Thank you. I want to first, in my side, say thank you to the Ministry of the Food and Security. And I want to add one other thing that made me worry a lot about everything that we are talking here is mostly you are talking for your group, human talk with our group. For example, why there is not anybody from Ministry of Health of the South Korea in this meeting? Why? And we are talking all the time that we have to work together. No. You are working for yourself, we are working for ourselves. I think that there is a big problem. We do not, sorry, from my part I am saying, we do not understand common work, shared work between human, between animal, between agriculture, between environmental, between all of them. And you had a lots of pellet, but this pellet was just for the animal. And in the end, sometimes add something to the GLAS. Yes, GLAS is a data gathering system that gathers prevalence of resistance. Just we are working on the how many of the pathogen is resistant for this antibiotic. This is very nice for the pharmaceutical company to make drug, but not for the human, not for the population. I think that we have to seriously think about how we can work together. Why just I am here? Why? There is no anybody from the human side. We have tons of things to talk with each other. And from your side, this is, and I see many countries, there is no any discussion in the country between these three parts. They don't know that there is or there is not. I think that it was not very good for the final discussion. Sorry.
Very quickly, please. We have to close this session before 2.
네.
So, thank you very much, Professor Nagabi, for your comments. Actually, for our part as Republic of Korea, we have our own AMR control and reduction plan, and we do have a collaboration between departments. Of course, there are many representatives, but they are not able to participate in. So, MOHW and the Ministry of Fisheries and the MAFRA, every participant from the relevant department are joining us here together. So this conference is only focusing on the foodborne antimicrobial resistance. So please ask for your gentle understanding. Korea, we do have a collaboration. So under the Infection Prevention Act, we need to establish a AMR Prevention and Mitigation Act. And last but not least, I do have one question to Dr. Garcinia. So, in farm system is a newly born, and this is a newly born global database. It has a great significance. So for Korea, food and agriculture sector generates a lot of data. So I do believe this data should be fully utilized in an effective manner. But in some countries, data generation can serve as a hurdle because of a lack of funding or budget. And also for the surveillance programs, they need to be representative, so they shouldn't be underrepresented, so that we can compare our data with the other countries' data. So for my understanding, from that perspective, are there any common strains or bacteria or antimicrobials for human purpose, or the number of samples? Are there any guidelines from the in-farm system? So I do believe this guide should be a prerequisite. So what is your take on this, Doctor?
Thank you very much for the question. So just
please answer about that within 1 minute.
Yes, I will. So very quickly, so there is the infant manual. In the infant manual, we have all the technical specifications, also recommendations on what microorganisms, specimen, species to submit into the system. And right now, in the stratified analysis we are doing with the data we have under the system, we will, of course, extract information that is most relevant for getting statistically significant epidemiological inferences from the data. So we will do that. Right now, it looks like the first epidemiological significant iteration we can have is in E. coli in broilers. So it might be more species and combinations in the future, but for now, in this first baseline report, we will start with that. And maybe just very quickly, but also to mention that I completely agree with Dr. Mosen on the statement, but at the same time, I also acknowledge the capacity of a human being to be interconnected with other human beings, and this also affects when we do our work. So when we do our work, of course, we work with our most relevant collaborators, but also a word of agreement, but also I think the world is also changing a little bit in that direction. We, for example, we meet with my colleague Jorge and my colleagues in WHO and GOHA on a weekly basis. We work together in integrated surveillance. Our systems are interconnected. InFarm is being developed with all the lessons learned from glass and through many conversations with the human sector. So we try to get this collaboration, but of course, will take time, will take effort. But thank you very much for the provocative statement, because it's very good for our thinking and for future collaboration. So thank you.
30 seconds.
Yeah. Yes, please.
Yeah, thank you so much. So I just wanted to give one further elaboration to what Alejandro said about the InFarm. So in the Asia-Pacific region, we have regional level colleagues, myself and Dr. Jutanath, who is also in the room, I think on the back side somewhere. We are the regional focal points, and if there are any questions from the Republic of Korea, we will be happy to support the country in better understanding of the system, which I'm sure you already had the training last year, the colleagues came, but if there are still some questions, we are happy to further support and discuss with you how we can take it forward. Thank you.
Sorry, we have 4 online questions. So, Dr. Garcia, you have 2, right?
I have 2 questions. Do I have time to answer them now?
Yep.
Okay.
1 minute per question.
So I have one question which is related to if participating countries in InFarm are allowed to use the data. So of course they are allowed to use the data. It's their data. They have access to a private interface where they can get access to their data, and they can also get access to the interactive visualizations that are produced with their data. And the second part of the question is if the reports are biased only on disclosed data. What is the role of undisclosed data? So this is a very good question, and it's a question that we were discussing also a lot in the inception phase of the system. We want to go in the direction of having 3 different levels of confidentiality, mainly because we need to progressively create trust in the system. We cannot go into a system that directly shows the identity of the countries in food and agriculture, because there are many other concerns. We are not on the human sector. We are here talking about other economic constraints and economic worries in countries, and trade is a big part of it. So that's why we need to go in a progressive way. The role of the undisclosed data is to be part of the system in the future, because we will continuously encourage countries to participate at level 3, so showing their public face. And then there's a long question. With the permission of the moderator, can I answer, or maybe I leave to Usman, or I can answer separately after the... I follow your...
After this session is closed.
Okay, yes.
And the next one is Dr. Jack here.
Thank you so much. I think the question is around, have there been any improvements in the surveillance systems once the funding was received, except besides the development of a surveillance system, the database, I believe. So yeah, so I'll give you an example without mentioning the names of the countries. As I mentioned in my presentation, FAO has a tool called Atlas, which is an assessment tool for AMR surveillance and lab networks. That was launched in 2019, and it has been applied in over 50 countries across the world, and over 270 labs, if I'm not missing any, have been assessed globally, including several countries in the region and several labs at the country level in the region, Asia Pacific as well. And the assessment is done, and it gives a country a score from 1 to 5, where the score of 1 says country has a limited capacity, versus the score of 5 says country have a sustained capacity, while the score of 3 says that the data is a reliable data from the country. So we have done the assessments in the countries, and several countries have gone through the follow-up assessments as well. For instance, a country had an assessment in 2019, they got an assessment again in 2023. Through the repeat assessment, we were able to see clear differences in how country has progressed their AMR surveillance system. And this is not only about data, it's about governance for AMR, it's about EPI systems for the AMR in country, laboratory or the data production systems in the country, AMR data and surveillance communication, and sustainability of the AMR surveillance. So around these 5 main pillars, we were able to see a clear difference in the countries in terms of their strengthened capacity, And several countries in the region, without disclosing the names, have achieved a stage of 4. Others have a stage of 3, while some are still at stage 2. But those who are at 2 were originally at level 1. But the results of these assessments are confidential. We don't disclose these results. We only share it with the country and the relevant partner in the country. Thank you.
Okay. And next, Mr. Maharujan.
Yeah, the question is how important of the AMR management consider. Definitely we are giving priority since we have conducted the passive surveillance since 2021. So the DFTQC, the government has given priority the sector, and we are the part of the NAP AMR. So there is some dedicated budget is already here. I just share that not the dedicated budget adequate in case of Nepal, but we have budgets for the annual program for giving priority to the AMR and residue surveillance. Yeah.
So thank you for the speakers and all past, no more participants. We're going to close this session, okay? I hope you have a safe journey back to home, okay? Thank you.
Now, I'd like to express my sincere gratitude to Professor Chae-Jong Chan from Chongping National University and 3 presenters for your hard work in this session. Please give them a big round of applause. So, 2-day-long conference was filled with many large turnouts, and speakers and session chairs and online and offline participants should deserve my recognition on behalf of the entire organizing committee of this conference. And poster session invites a lot of presenters. Thank you very much for your participation. So in this poster presentation, so AMR characteristic study and AMR residual detection methodology and Codex elementarius application, and there are 15 posters presented. So all the presentation materials are available on our official homepage. So if you are yet to finish your survey, so for betterment of the next year's edition, please your inputs will be highly grateful and appreciated. Next year, 6th MFDS GCFA is not planned in September, so 2026 May we will have 6th edition of our conference, so your participation and interest will be highly appreciated in advance. This brings us to the end of the 5th MFDS Global Conference on Foodborne Antimicrobial Resistance. Thank you for your participation once again.
살며 살아가는 행복, 눈을 뜨는 것도 숨이 벅찬 것도 고된 하루